Very good morning and a happy Holi to all of you. Uh, my name is Manas Shukla, and unlike all of you, I'm not a faculty. I'm actually a student. I'm currently I'm applying for my PhD applications abroad, and I was in the lab of Dr. Vivek sir, which is in Systems and Computational Biology Department of the School of Life Sciences. There, my work was on characterizing the population genetic metrics of the endangered vulture species in India, and we've mic is not working. Hello. I might have to, I think, take it in my hand and then talk. Yeah. So, yeah, that was some brief discussion about my work. But my lab majorly works mostly on the fields of genomics, which is reading and generating inferences based on the DNA sequences and then further transcriptomic and proteomic sequences. But we've used a lot of machine learning pipelines in a lot of our works. And here is one example. And although this lab session should have held a little later than the earlier theory sessions, because I'm told this is the first session for life sciences. So I'll try to keep it a little more generic or a little more theoretical, and then we can start doing on a lab work problem, which would be very short. I have generated a short data set for you to work on, and it would be easy. It's just based on graphical user interface where you will have to log into a website and then upload pictures and see whether it recognizes correctly the species which are there in the picture. And yeah, without any further ado, I think I can start. Yeah. Before starting this lecture, I'm assuming you already might have been listening to a lot of talks which talk about the major algorithms that machine learning has to offer. And there are literally just two main branches, and then the others are learning methods which are drawn on from those two. The first two are unsupervised type of machine learning and the supervised type of machine learning. And you might already know the difference between them. In supervised type of machine learning, there is data which is already labeled. So you already know what your input and output are, and you can actually generate a feedback system, because once your model gives you wrong results, you can actually tell it, okay, the result was wrong, and you can correct based on your mistake. And the this was supervised learning. In unsupervised learning, like the data points, in unsupervised learning, this is what happens. In supervised learning, where you have to separate data points based on no labeling that has happened predetermined. So all the data points look exactly the same, and you to try to find out some inherent structure between those data points to see if they can be organized into different groups or clusters and things like that. So that's supervised learning. And sometimes we have a mix of both of these data sets. For example, we can have some data set which is labeled and some data set which is not labeled. And when you do the approach like that, then it's called semi-supervised learning, where once you clear out the supervised part, where based on what categories could be effectively distinguished, you filter out those, and then you do the unsupervised learning part, where you eke out the structures between the inherent properties of the elements which are your data groups. And reinforcement learning is another kind of ML approach, which learns from its mistakes. For example, how you would teach a robot to learn something. It would learn from the environment, it would learn from the outputs, and then it would inculcate in all of its calculations and do some other things. There are, some ensemble methods that also come in under reinforcement learning. For example, you might have learned about positive reinforcement and negative reinforcement as concepts in biology, where okay. So, for example, uh, you've all seen children in normal walks of life, and there are some times when parents try to discipline them in different ways. For example, if someone is not acting in a way that you want them to, then you might associate them with something negative. For example, you might ask them to go and stand in the corner, and then they associate that feeling with standing in the corner, and then they think, okay, this is something bad, I should not do this. So this is a negative reinforcement. With positive reinforcement, you would appreciate the good activities. For example, giving out chocolates so whenever someone does something good. So that's positive reinforcement. But yeah, that's topic for another discussion. Someone would definitely talk about ensemble methods and reinforcement learning algorithms. But this is the gist of it. And here it's a lot of different subsets of all these types where different algorithms approach problems differently. And there are some general applications written over all of those bubbles. For example, in unsupervised learning, the major backbone of the whole approach is clustering based algorithms. They cluster based on the properties of the data, which is not very apparent or conspicuous. And the other part is dimensionality reduction, where a data set which is in lot of dimension, they squash it together, they squeeze it into lesser dimensions so that it becomes easier for visualization and noise cancellation and a lot of other things. And yeah, it also decreases the computational capacity and power that's needed for each step. And the other type of learning, which is supervised learning, which is the most extensively studied mechanism, so not like a mechanism, but a ML algorithm, which is majorly grouped into two classes. One is classification, which takes into logistic regression models, and the another is linear regression model. So in linear regression models, the values would always, the values can go above 1, even though the whole function would be ranging from 0 to 1. I'll show one graphic after this one, so you'll understand it better. But yeah, in logistic regression, there is a classification, solves a classification problem. So there has to be two categories, for example, a yes or a no. For example, a mail arrives in your folder, in your email account, and then some classifier is built on that classifies whether that email could be a probable spam or not. So there are two categories: mail being a spam or mail not being a spam, and then it takes you into your spam folder where you see a lot of spam mails. So that is based that kind of thing is based on the classification problems, where some other kinds of work in normal walks of life has also been made convenient. For example, customer retention or the diagnostic problems and image classification, some of the things that classification covers. And regression, because you can expect a trend from regression. It establishes there has to be a linear relationship between the dependent and independent variable. So for example, if your height is increasing, then it's obvious that your hand size would also increase. So those two have continuous properties. The temperature is a continuous variable. Age is a continuous variable. So in one simple example, if you think the linear regression model can tell you whether a student would get 1 or any number in between from 1 to 100, while your logistic regression model would tell you whether a student will pass or fail. So something like that. And it's also used in a lot of other forecasting problems, for example, market forecasting and people segmentation, population growth prediction, a lot of other population dynamic based simulations. Yeah. So this was what I was trying to tell you through the linear regression and logistic regression models. So here you see the predicted y range, which is the dependent variable. It would depend on the independent variables, which would be a lot of these points, which are shown here and there. So, but it would only be linearly related. So it can only hover around this straight line, the relation. But in, the straight line, but it can go over 1, it can go below 0. But in logistic regression problems, you would always get two categories, where one line would separate the other category from being a positive and the other automatically becomes a negative. Yeah. Here, these are some other types of supervised learning algorithms that also, classify some other things and tell you what would happen. So I've already talked about linear and logistic regression. Support vector machines is one another kind of example, where imagine that a lot. Can I use markers? I think. So imagine that there are a lot of points here, lot of points, and they are indistinguishable. And you have to draw a plane which separates the line, which separates a line and the points from an equal distance. So that should be an equidistant plane. So and here in support vector machines, each of the point or the dot, are you not getting it?
Use the lower voice.
Okay, yeah, it would be better if I had a marker, but yeah,
Marker is there.
this one, this one's not working. Yeah, anyway, so I'll show you in form of another diagram. This is meant for something else, but I'll show you just for example. So imagine that these are all same points. There is no red triangle, there is no blue dot, circle, or a pentagon or a yellow box. These are all same kinds of points, and they have some inherent property that they could be pre-labeled. But what this algorithm does is it will try to sketch a line which separates all the points from equal distance of the line, and that line would, yeah.
Make it be not visible from.
I'm sorry. So anyway, so you see an example here where two types of data set points are seemingly being separated by one line here. This one line in 2D plane, it would obviously be a line. In 3D, it would be a hyperplane, and which would separate the other 2 points in another dimension, 3D dimension. And here, the objective is to also assign margins or other lines. Here, you see, this line and this line, they would be equidistant. This line and this line, they would be equidistant. But assigning a margin here would mean that this data point incorrectly comes into here because it comes in the margins place. So it could also be classified in another iteration as this data point, not as this groups. So the goal here is to keep the margin maximum where no data points fall into. So support vector machines work like that, where each data point would have its individual value, and they'll try to, based on the distance metric, it could be Euclidean, it could be Mahalanobis, it could be any type of distance metric. Based on that, they group points together in one group, draw a hyperplane, and separate that with the another group.
Say the dotted line and the solid hyperplane. So those are not considered to be in that group.
Yeah, this is very interesting. I mean, I'm assuming that you're talking about a spherical data distribution.
Good data.
Yeah.
If you have a solid line,
Yes.
that is your hyperplane.
Yeah.
And you also have a dotted line.
Yeah.
What does dotted line mean?
Dotted line here would tell you how far off the prediction, how far off the line here. I mean, because the approach here is maximized in such a way where this line is also kept at a maximum from the other group and also kept maximum from the other group. That's how it would tell you that there are two distinct groups. The more the distance between the two types of data points, the better the group identity.
So if any data point is in between that dotted line and the solid line, so would I consider it to be sort of outlier, or it can have sort of chameleon nature? Both can be possible.
Yeah, because this is obviously a machine learning algorithm, it can give you wrong and false results. So the later case can happen, the previous case can also happen, but there are certain tricks to, optimizing how you would draw a hyperplane. So you can tweak with your data first, remove some of the noise first, and then do the hyperplaning thing here. And also, this is not just true for this kind of thing. For example, I'll show you, just imagine that there are spherical distributions. Not there, okay. Spherical distribution meaning one type of data points are here, the other type of data points are here. How would you draw a hyperplane then? Because it's not possible for a line to be there in a spherical distribution. So what they use is a kernel trick. They do that, they increase the dimensions of the data altogether by mapping it to a ReLU log function. And when it happens, you imagine that this thing is coming out, it's another axis. So even though this was a concentric circle here and this was a concentric circle here, this could be a little higher than this one. So in another direction, in another plane, which would be coming out of here, the 3D plane, you can separate those two spherical distributions. Yeah. There are other classifiers as well, but those are probability based classifiers, and they will be talked about a lot in later classes because forecasting based talks are scheduled. And yeah, another example is a decision tree. A decision tree, like any other thing, starts from a node, and at each node you would have two choices, yes or no, and then it goes on to the another node, and there it splits as well, yes no, and then yes no, then yes no, and then yes no. So it can tell you a lot, in simpler terms, in sketching less algorithms, how you would be able to do better on a given day. For example, if you have to go play golf, you would have to see whether the situation is windy or not, whether it might rain or not, what types of cloud are there. So decision tree can help you make those decisions, and these decisions can also be included with other types of classifier to make the results more robust. Yeah, these are some of the actual can I increase the hello okay, is this fine? Yeah. So these are some of the actual applications of against the algorithms which are written here. For example, random forest is used in fraud detection and medical diagnosis purposes. So this has other types of methods, boosting, bagging, and other method as well, which would be a talk series for a topic for another series or another lecture, which is going to be taken to you by, I think, Vivek sir. And yeah, and linear regression can tell you about forecasting, and trend analysis, how would something change over time. And support vector machines would also help you with classifications. Decision tree doing the same thing. Neural network, which is one of the main topics or hot topics of today's world, which is also one topic in the talk that we are going to do today, the lab work. This helps in image recognition, which is going to be forming the basis of our lab work or demonstration. And yeah, it can also take cues from sound. So from sounds as well, if there is enough data, if the data is not noisy, you can sometimes build a classifier which is robust. Yeah, so this is the kind of learning that happens in unsupervised learning. So here, because data is not pre-labeled, they try to find if it has inherent sometimes of structure. So for example, imagine that they were looking like similar dots first. So this method here is called centroid-based clustering. There are other clustering methods, density-based clustering, centroid-based clustering, hierarchical clustering, which is shown right next to it. And here, what it does is, depending on how the points are scattered, you would assign a k value being your number of centroids here. So it would, according to the point distribution, it would assign centroids randomly here, 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 and then it would calculate the distance, the mean distance of each point to the centroid. So for example, this point here, whether it's closer to this one or closer to this one, this would be found in the first iteration because the centroids are assigned randomly. So what happens is, at every next step, this expectation is maximized by improving our choices, because we already know, okay, what the distance of the data points to the centroid mean was initially. So we can change those once we know, okay, this was far off, this point should not be, sorry, this point should not fall into this. Then over multiple iterations, you would find a pattern where where a clustering happens, which separates the data based on inherent properties. And yeah, this another type of clustering is kind of important for taxonomists because it gives you a nested grouping structure where you would see relations of one particular group with respect to another if they share some inherent properties. For example, the animal kingdom here, all of the animals, vertebrate, invertebrate, they would have some common features in all the animal kingdoms. Vertebrates would be a little different from invertebrates, but in general, animals would have some common features. So under Animalia, you would see two types of different clusters, one that would belong to vertebrates and the other that would belong to invertebrate. And that's called hierarchical clustering because there is a hierarchy. For example, just like that happens in biology, kingdom, phylum, class, order, family. Yeah. And yeah. This is one kind of illustration that I got from a Nature review paper, which summarizes how some of the approaches in biology are currently making use of AI/ML models. And the first thing that this illustration talks about is the advent of the deep learning technology that has happened. So with deep learning technology, it's a part of a neural network thing that I will talk about later. Here, it has made a lot easier for people to discover drugs. At the time that it used to take doing homology modeling, threading, and then docking and everything else, it could be minimized to a lot less extent using these algorithms. And we have seen with AlphaFold being the predictor of protein structures. It takes up the sequence of DNA, and then it will model based on the DNA how the protein would look like. So predictions, any mutation impact predictions, for example, how a variant could be related to a disease. And in precision medicine, because everyone has a different environment they grow up in, everyone has different susceptibilities, different types of markers. So those susceptibility could translate into a genetic risk, and these AI models can also help you find those. And another type of work that they do is structural variant prediction and variant annotation. For example, a lot of you might have worked with SNP files, SNP markers, which is contained in the VCF file. So these algorithms can itself be used. For example, CNN here, which would be talked about later, convoluted neural network. It can be used to group variants into one particular group. So, I mean, variant calling can be improved with the other pile-up methods if you incorporate machine learning algorithms. And you might have seen some of the posts recently being circulated on either Twitter or other platforms where you might have seen one picture where there is small clot or blot in lungs of one particular guy, and that clot was identified by some AI learning platforms to be becoming a tumor. And that was a prediction that cannot be made possible in the normal labs, because at that point, I mean, in the present time, that blot was still not cancer, it was still not benign, but it was something that the algorithm had learned from a lot of different images. It had taken in a lot of MRIs, a lot of X-rays, a lot of other things, and then it recognized, okay, when this kind of pattern emerges, it is most likely going to give rise to a tumor. Then that's what the prediction was, and that's how it came out into the light. And some other DNA sequencing based methods also exist where improvement have been observed when machine learning algorithms have been used. For example, a functional annotation of non-coding DNA, even though most of the DNA is coding, non-coding, and it was previously being called as junk DNA, but now we know it has a lot of other functions in terms of expression and other things. And you can also map pathways starting from a sequence, the KEK pathways that we know of, and the other things. They can easily be mapped out if you start just from a sequence and have a model which integrates different types of models and would give you the result. And the docking thing, whether a protein would be toxic or active, these types of activity classification things, they are really very, very easily, doable with machine learning algorithms. And yeah. This particular section here shows some other emerging fields. For example, one very hot field currently is the emergence of multiomics. So people have been trying to use AI/ML algorithms, the integration of those algorithms in multiomics, and see if their predictions could be better, if they could be made better. And spatial transcriptomic is another domain. Similarly, synthetic biology design is also one where integration of AI/ML is being looked into. But looking in general, why would biologists want to classify, or why would biologists want to use AI/ML algorithms when a lot of different alternatives exist? Is because we work with a lot of data. I mean, sometimes those can not only be computationally expensive, but also they would lead to overfitting issues, and they could give you spurious results based on how good or not good your particular data is. So here, um, to interpret things better under a less computational power and to analyze things quicker, some of the AI/ML algorithms have been routinely used, and they are more in vogue now. Yeah. This is some of the other applications that I think I did not talk about, 2 or 3. Even though I talked about the imaging application where whether some disease could be picked up from an image, that can happen, genomic and proteome analysis and drug discovery. But there is one also very important thing that happens routinely. You might have heard about all the endangered species tracking Zoom. Yeah. Yeah. So actually, we have talked about these 3 applications, so it's fine. But some of the other things that actually are being used in real life applications are, one thing to determine whether a plant would develop a disease, looking at the leaf patterns, looking at the rust patterns, looking at the discolorations and the texture of the leaves, whether a plant is more likely to be, bad crop that season, how would its yield be, and other things. But you can predict diseases also, and it's actually being used. And the other thing which routinely happens is with camera trap monitoring. For example, it's very hard to count how many particular individuals of a species are there, especially with the lack of resources that we have. But what is routinely done is camera traps would be placed randomly in the areas where higher chances of finding endangered animals would be, and then those camera would record pictures randomly, and based on the huge picture data set, some image platforms, AI based image platforms, they'll try to recognize from those images which species it was. After recognizing which species it was, it will also look into the other photos of the same species and recognize whether it was the individual which had been encountered once again, whether it was the same individual or a different individual. That's how they generate the number or the census size of a population as well. Yeah. So talking on the same lines, there is actually one particular platform which is called the Journey Bio Click 2. It was an initiative started early, and this particular database has over 200 million images, including a lot of species, vertebrates, invertebrates, and this particular database, because it's so huge, it could learn patterns on its own depending on what the input tweaking has happened. And one very interesting example from it was it because it had a lot of pictures, it analyzed how Darwin finches, the finches that you had studied in when Darwin encountered those in Galapagos Island. So they had different habits. They had, some of them were eating fruit, some of them ate insects, some of them ate crops. So their beak sizes were different based on the images which showed the beak sizes and their pattern, where it was sharp edged or not. The algorithm actually uncovered some of the patterns which were actually not known beforehand. And for a lot of different training data sets, I mean, whenever you would, if any one of you would ever work on a species training data set, this would probably be a source that you should look into, because this contains the most curated number of species and also most pictures of those species as well. I'll talk about some actually real problems that we had faced in our labs. This was a work that that our lab had decided for a master's student. She was my classmate. So what was the problem is, I'll initially talk about what in general the problem with Parkinson's disease is. So here in this particular graphic, you can see 4 particular phases being shown. One the risk phase, one the preclinical phase, one the prodromal phase, and the other is clinical phase. So in risk phase, can be counted as what happens to your environment, what happened in your environment, how your environment had affected you, and what is your genetic susceptibility to a lot of markers and things like that. So you are born with things like you are either born with things like that, or you gain things like that during environment, during your growing up in the environment. So risk phase is not accompanied by a loss of dopaminergic neurons, which actually happens in Parkinson's disease. So there is a particular region called substantia nigra. It's responsible for a lot of motor activity and also reward-based systems. So their specialized types of neurons actually help them do those. Those are called dopaminergic neurons. So their degeneration happens. And what actually happens in the preclinical phase is when even though symptoms appear, not symptoms exactly, but even though markers are there, even though onset of a particular disease has started, there are no symptoms. So currently, this is a phase where no symptoms are being shown, but it has the neurodegeneration here, as you can see from the levels, it has already started. So neurons are already being dying, but we do not have seen symptoms in people in the preclinical phase. The other phase is called prodromal phase, where some of the soft symptoms appear, and they tell you the they, I mean, they are some of the things that would make you susceptible to developing PD more. For example, people who have inflammatory bowel disease, people who have REM-based disorder, RBD disorder, and people who clinically suffer slight motor symptoms or olfactory dysfunction or even depression sometimes. So these are some of the things that would make you susceptible to develop PD more. And but still, according to our clinical classification things, we do not consider them as markers for PD. Only when almost more than 30% of the neurons are lost, and when the symptoms are in their full fledged, doctors realize and clinicians realize, okay, this is PD, and it does not get treated beforehand. So neuron degeneration has already kicked in, neurons have already been cut out, shed out, but still you do not have PD according to clinician diagnostic criteria, whichever is set up right now. So this was the problem, and what what my lab thought how it would be easier to tackle this was, building a classifier system that tells, based on the gut microbiome of people, because it has already been studied that people who develop PD, they have different repertoire of gut microbiome. For example, bifidobacterium and lactobacillus, they are high in number in people who develop PD, while faecalibacterium is less in number. And a lot of these things are correlated with the prodromal marker phase. So our idea was to classify people into control, in prodromal, and in PD, early PD. So that would mean people could be classified into groups where even though symptoms were happening and they were not being regarded as PD patients, they could still be diagnosed as PD patients, just based on the onset of those symptoms, just based on the markers and the composition of the gut microbiota, sketching a causal relationship between those two. So that was the plan. Yeah, gut microbiome dysregulation and dysbiosis that happens. Even probiotic have been shown to have some effects on the development of PD. People who take probiotics a lot, they have been shown to be somewhat susceptible to TB than people who normally don't. Okay, yeah. So overlapping symptoms, for example, these are all some other kinds of diseases. This could be due to other diseases, but one thing that almost 32% chances increase of you developing a PD if you have this particular type of disorder, REM sleep behavior disorder. So there are some close associations. For example, there are some prodromal markers which are associated more with PD. Inflammatory bowel disease, Crohn's diseases. These are some of the examples which are associated. So people who have a lot of these prodromal markers and a gut profiling that tells, okay, this could be the gut profile of a PD patient or an early PD patient, they would be classified accordingly, and they would have a higher chance of developing PD. So this classifier works in that way. REM, rapid eye movement. So rapid eye movement. So during sleep, there is a phase when you might have seen even your children, or you might have observed other ones. So they do like this. Their eyeballs would rotate. So that is the phase right before you step into deep sleep. That is when your eyeballs move and you are just going in deep sleep and a dream. CSF, cerebrospinal fluid. Yeah. So yeah, because these do not have specific markers, no markers have been validated for PD yet, and to I mean, effectively say that a person would eventually develop a PD, you would need markers. And because those have not been yet diagnosed, this is why some of the algorithms are using AI-based models to see if the prediction can happen. No. I mean, the feature extraction is itself a process in all of the machine learning based algorithms where you would map a particular function. I mean, based on certain kernels, based on certain filtering criteria, it would recognize some features and it would build on those features. So here, I mean, it's not exactly that numerical values are associated. I already talked about the classification problem using logistic regression. So, but in logistic regression, you heard that okay, there could be two categories, yes or no, and I mean whether someone would pass or fail, whether it would rain or not. But here it was a classification based on three systems. So using another kind of a function, you could map into 3 groups, 4 groups, 5 groups. So those are multi-class classifiers. So you could build binary classifiers, those are easiest, but multi-class classifiers are going to be more robust if you are going to classify into more groups. Because as we have seen here with PD, the progression is continuous, but the phases are a little different. I mean, a preclinical phase contrasts a lot with prodromal phase.
really binary. REM,
No, this is not a marker, no? No. REM, I mean, exactly is not a marker that you would associate from a genomics perspective. It's a marker that would tell you what would happen in a person. Marker of the chances of developing PD. So it is not included rigidly as a variable, but included as a dependent variable on a lot of other things. Because it's just not one particular feature. I mean, features from other groups would be extracted as well, and then they would be pulled into the model. Things would become a little more clear when I talk about this CNN model, which would be used in the image recognition thing. That should be the topic of today's lab. Yeah. So the talk had detailed that we should have used two kinds of tools. One is called the iNaturalist and the other is called Wildbook AI. So I'll tell you what each of these do, but they are heavily curated. They are heavily in collaboration with people as well. You can open up projects, you can add people up in a project, you can ask them to contribute. So what happens usually is it's easier in this sense whenever you go out and you see something interesting in terms of a plant, in terms of a spider, any kind of an animal, you can snap up a picture. Once you snap up a picture, it's not very likely that you would know most of the species. So you can go search that image. I mean, after you have logged in to any of these platforms, you can upload the image, and based on the already curated dataset that it already has, because a lot of people do the same things. A lot of people have deposited images. So there are over 10, 20, 30 million images, and some other projects have worked extensively as well. So there are different types of images. You can filter through a particular region. For example, if you want to look into how many reptiles are there in Andhra Pradesh, you can filter through Andhra Pradesh and type reptile and see how many photos have been recording during a time frame of a reptile. And a lot of these are not very annotated because people are required to also put up classification of their own. That's how it makes this particular platform better. So if you know for sure that this species is this species, for example, if you know that this particular species of spider is a jumping spider, and you write there it's a jumping spider, the suggestion from AI would also be something similar. You would, even before typing jumping spider, you can get a suggestion for a spider, an arachnid of the same class, or a jumping spider. So there it would go over to different people later, and they can comment on your observation. They can say whether you think this is a jumping spider or not. But apart from that, the model gives you a score. I mean, it predicts you a score, and based on that score, it would tell you anyway whether the image that you have identified as this is a high, I mean, is a good prediction or not. And the Wildbook AI does a similar kind of thing. I'll tell you how do they eventually reach up to, I mean, recognizing the features from an image. Uh, so yeah, the foundation of all the image recognition systems that happen throughout are based on convolutional neural network, which is one class of neural networks. So here, what happens is there are I mean things which are studied in layers. Layers meaning in it would extract out a few feature points. It would give a filterable kernel for some different criteria. For example, texture, edges, color. So this would be the starting most simple variables. And starting from edges, color, textures, it would build on higher, deeper features. And in each layer, the information is connected and it would be transferred to the other layer so that it learns better. And during this multi-hidden layer transformation, the data is curated nicely and eventually you would get a higher prediction result. For example, this particular graph that you see here. So this is your input layer. Input layer meaning the first layer that would tell you, okay, this particular thing is an edge, this particular thing is a line, and then from edges and line, you can see, you can connect other things. I mean, this is one particular hidden layer, this is another hidden layer, this is another. So this information would reach back to this, and it would reach back to this, but not all of them would be deeply or densely connected. Based on the connections, this can be made very robust or not. So what happens is obviously the first level of recognition after a color and lines, they happen, then the textures and shapes are recognized, and textures and shapes, they would be pooled on together to recognize whether it looks something like a cat's ear or a dog's nose or something else. So then the next part of prediction is object parts. So from those textures and shapes, it would predict parts, and based on the parts, it would predict the species. So this is how the whole transformation happens. But I mean, as simple as it sounds, there is some mathematical, I mean, nuance in this. So what happens is there are two types of layers. One is called convolution layer, and the other is pooling layer. So in the convolution layer, this kind of thing happens where edges and lines and these things are recognized, and in more deeper levels, more extensive information is processed. So this particular thing is sketched to a non-linear transformation, because for these kind of recognition, you cannot just gather on linear relationships between data points. So it is mapped to a non-linear function so that the model can learn more. Non-linear function meaning a lot of the, I mean, characteristics of an image would not be linear in the sense they would not depend on with a straight line on another dependent variable. They would not vary with a straight line. Yeah. So that happens, and the other layer that I talked about, that was the pooling layer. So there, to reduce computational efficiency, to improve computational efficiency, and also to reduce time for the computational efforts, it squeezes the data into less dimensions. So it reduces the height and the width. So less computational power per pixel is needed. Because images have a lot of pixels. And a high resolution image can give you out of bound error in terms of memories and other things. So to reduce the computational cost, this is done to squeeze out the data into lesser dimensions. And then it's mapped to another mathematical activating function, which is the soft bang function, I think. Yeah.
Traditional neural network.
Yeah, it's actually a good question. So, a lot of times, the thing that I showed you here, their classification and feature extraction happens in the same process. I mean, during the model development and building. So, feature extraction, meaning the edges, lines, contours, colors, that, and classification, whether those make up to a particular part, cat's eyes, dog's nose, peacock's particular uvula, anything. So those two happen simultaneously, but in different types of models, they can happen either before or after. So you can extract features before, build on a particular classifier, and then run the learning algorithm where the classification eventually would happen. So it actually depends on when a particular feature is being extracted and how it's being extracted. But to talk in depth about neural networking, including deep learning models and others, it would need probably 2-3 more lectures. And I don't know if you have talks on neural network. Do you? Okay. Okay. Anyway, what do we have time for the lab activity, or how much time do we have left? We have? So we have almost 1 hour or half an hour? Half an hour? Okay. Half an hour is fine. So yeah, this is the overall result of both of these algorithms. So with common species accuracy, the common species accuracy recognizing species which are not endangered, those are commonly known to people. It has an accuracy of over 90%. And the species which are endangered or less in number or threatened, the accuracy kind of drops down to 75 to 85%. And another, yeah, it also shows you the top 3 to 5 prediction for each input, and the processing time is very, very less, as you would see in your interface when I'll let you use the data. But there is another type of problem that I would also like to talk about. This I would tell later. Okay. Yeah. Anyway, so in actually my master's project, we could not get sample permissions for blood or tissue samples for the vultures. So what actually happened was, because we had very less time, we had 1 year, we collected molted or shed feathers from 2 vulture cohorts, one in Nehru Zoological Park and the other in Rajasthan. That's in Bikaner, Zorbir Conservation Reserve. So from different individuals, feathers arrived, but because those were shed on the ground, we had no way of knowing whether it belonged to this individual, that individual, or whether 2 feathers could be from the same individual or not, things like that. We did not know anything about the individual identity and the species and identity as well, because we collected only feathers. We could not even pluck from the individuals which were present. Those were the shed feathers. So there became a problem of having unannotated kind of data. For example, you would have a lot of pictures on avian fauna and avian species, but those would be pictures that would show you almost everything, but you would not extensively have a database for feathers. I mean, you would not have a database that would show you feathers of harpy eagle, that would show you feathers of bald eagle, or feathers of other accipitry forms, for example, vultures or any other things. So a lot of researchers face this type of problem where the data is not trainable. So what happens in those cases? What can be done in those cases? So actually, it would require a mix of, 2 worlds. One is, this is leveraging existing collections, because you cannot do anything about the database. It's not like you can generate a database of feathers in 10 seconds or 1 year, and that would be expensive, because training models requires a lot of data. So you use already existing data. I mean, a lot of you would focus on birds if you have to recognize a feather. So you would focus on birds, and from those birds you would do targeted image collection. You would focus your algorithm such that it does not take into account any other feature than the feathers. So it would account only for features, and then you would try to train your models based on the reduced representation of the database that you already have. So in such cases, you kind of have to do a trade-off where even though you do not have annotated data, you can try to approach the problem in some other way. Yeah. Yeah. This this was the data set structure that I had prepared, but I don't think is a way for me to transfer you files right now. Um, is there a common group email ID, or there is WhatsApp? I can probably send you data on that. So the data, I mean, in itself would look something like this. It would have names for each, image here. Then metadata CSV would contain all the types of information about the image that is available. For example, when the image was clicked, what was the latitude, longitude, where it was clicked, and whether the species is, I mean, what do you know about the species, whether it's alive, whether it looks diseased, whether, I mean, other kinds of comments that you can make. So it's curated heavily by people. Yeah, so this is one example of what could be present in the CSV file. Image name, timestamp, latitude, longitude, camera trap ID, nodes, these kind of things. So yeah, I would send you, I mean, a zip file that contains images of the order accipitry forms. So that contains eagles, vultures, hawks, kites. All of those individuals would be there, and because it's temporally collected over a spatial, over global spatial resolution, it would have individuals from different countries. So what we will do, we'll try to input one particular picture and see if the image data set recognizes it as correct. Yeah. So I think I should start the demonstration anyway, and I should send you the files. How can I send you the files on WhatsApp? Hmm. Drop in your number. Send. Send you this. If you people, do you people have access to internet on your desktops? Okay. So what you can do is, because this would require you to log in or sign in with an email ID of your own. So you should go to this website called inaturalist.com. Sorry, .org. You can type just iNaturalist on Google bar, and the first result that comes up would be that website. So once you have logged in, the interface would look something like this. There is home page, there is profile page, there is observations page where you would make observations. But I'll go a little slow. Once you've all reached here, you should first log in to your account using a Gmail ID or any ID, email account, and log into this website. And once you have logged in, let me know. Yeah, this website. Yeah, iNaturalist.org. If you don't mind my asking, yeah, you have to provide your email ID. Yeah. Yeah, this is also one way. Yeah. Not login. You will have to sign up first. Yeah. So write an email, a username that's not taken. That depends on.
Own username.
Yeah, username. For example, if Sridevi, something like that.
Password.
Password you have to decide on your own. It would be your password.
What is the username?
Username could be any name of your choice. For example, it could be your name, but let's say if your name is taken, then you would have to
Not mail ID.
go to sign up, not login, because this is the first time you're doing.
Sign up.
Then enter email.
Mail app.
Yeah, and then yeah. You can write your own name. If your name does not work, you can write some number after the name. Then you have to choose password on your own. No. Password would be the one that you decide. No, it does not. It should be more than 8 letters, just that. Make a password of your own. You can try using, whatever passwords that you use for normal things, you can make a password here. But you can also save it here. Once you log in, there would be an option to save it to this desktop. You can either or choose to or not, but do not, because this is a system laptop that would be here. So Yeah, I shared to Mr. Srinivas, and he should share to all of you. Huh? Defense?
WhatsApp is open.
No, you already have an account.
Okay, he's downloaded a phone. I'm asking him to download it.
Oh, okay. The data set. So anyway, I'll show you a quick example first. So should I start the demonstration? Okay. Okay. If you don't mind, you can look up here in the screen. It is the first thing that I'm going to show you is how to filter through the data set to get what you want. For example, does anyone have a particular preference? Do you want to, I mean, look at turtles in South America? For example, if that happens, we can just write for species. Here we can write turtle, and then turtle and tortoise, it comes up as a recognition. So turtle and tortoise is a group. Click on that, and then we can look at places. America. And then here North America and South America would come up, or the US would also come up. We want to, let's say, look at the South America, and then there are further filters that can help you dwindle down the number. So let's say, for example, if you want to look at the turtles or tortoises which are only in, huh? Yeah. So yeah, come to in the top bar. Uh, Whoever has logged in the top bar, can you see 1, 2, 3, 4, 5 sections? So in the identity section, click on that. Yeah, identify. Click on that. And then you can see two particular bars, right? One where I have written turtle and tortoises, and the other where I've written South America, right? So you can choose of your own accord. You can choose for any species. Yes. Huh? You're not audible. It's not opening. What?
Yes or no?
Yeah, it's fine, it's fine. It but oh, okay.
Should confirm in our mail.
Okay, yeah, there should be a confirmation on your email. Yeah, click on that.
Where to confirm?
See now, I think you can search.
Email confirm. Confirm mail ఎక్కడ
No, send confirmation now, and now it would come now. Go back. Yeah, just click.
They're asking how to scan the image.
How to?
How to identify the images.
Images. Yeah. One second. Okay. Anyway, so because we have less time, I would ask you to only look at the screen for some time, and you can try on your own, because this is very, very, very easy. The only one thing that you need to know.
Ma'am is asking how to identify images.
Yeah, I was going to do the same thing, but I have to show demonstration to everyone. No. Yeah. Huh?
It is not yet.
What?
Demonstration is not done.
No. It would take just 10 minutes, but people would have to look at the screen. Okay, so everyone stop doing whatever they are doing. So I'll anyway transfer you the workflow, whole workflow as to how you would go through the logging in and everything else. But look at the demonstration here first. I mean, as to how the tool works. So you would anyway, once you log in, you can, I mean, discover on your own. You can discover some sections. You would know things on your own. But let's see what the tool can actually do. Okay, so. The first thing that I wanted to look at was can it identify or can it annotate and tell me how many turtle species are there in South America, and then I can apply further filter. A further filter meaning I can write up a date. I mean, how many turtles were observed on a particular date in South America, or I can write up a range. I mean, how many turtles were observed in that period. I can write whether the vultures that it has to show should have been a captive individual or a threatened individual, or the entry should have had photos or sounds or any video recording. So things like that you can tweak here, high or low, in terms of your priorities. And it further has more filters, so you can even look at some users particular project. So I'm not applying a lot, I'm just updating search filters here, and then I just search. So once I once I do the search, whatever filters have been applied, they would culminate into this result. And so now you can see there are over 138 tabs here, 138 tabs, and in each of these tabs there would be at least 25 species, 25 particular photos. It could be same species, it could be different species. So this is how the intense collection looks like. I mean, belonging only to turtles in South America, this is how many, which is documented in this particular platform. And coming to image recognition, so yeah, you go to your observations, and once you go to there, I mean, this is just one I was trying for, I mean, checking for this image. You can do for another image as well. So here there would be a section called upload. So in upload section, you can just go click on there, and it would give you two options: either choose files, or more import options that also gives you CSV file thing. So if you want to, for example, if you are a user and you know, okay, these are the species and I'm confirmed, okay, this is my best effort and this should be the species, then you can directly have a zip file which contains all your images, and a metadata says CSV file which tells you all those things that I showed you in the image format: timestamp, latitude, longitude, and other things. You can directly upload those and it would create a project for you. I mean, it would have everything as annotated for the different people. So but that's another purpose here. Let's say your purpose is to recognize I mean, a snapshot of a species that you just captured. So how would you do that? Here just go to choose files and choose any file, I mean any picture that has okay, I think I haven't downloaded that particular dataset myself. 1 minute. I'll randomly download an image from Google and then check if we get the same result. So I have downloaded a particular picture of an Egyptian vulture. Egyptian vultures kind of look like this. Uh, this is what an Egyptian vulture looks like. Let's see what the platform gives us as a result once we drop this. So this is the image that we uploaded. After upload, it would show you three particular sections that you should, input most information in. But here, before writing species name, just click on here, and it would automatically load suggestions. And you see the first suggestion that pops up is what? Egyptian vultures. So that's how the identification program here would work. So you would drop in one particular photo or even a collection of photos, and then just on the species name, you do not have to write anything. Just click on there, and it would automatically load suggestions for you. You can choose any particular picture of an animal or a plant that is available online. You can download it and then check for it. So this is how it would tell you, but this is just not one application of it. What you as a user should do is input more and more information, whatever you are confirmed about. For example, here it asks you the species name. If you were not confirmed, you would you would not write the species name, but you can write the date, you can write the location, where the species was spotted, where the date was listed, and there are other tags as well. For example, if it's a part of a project, or when the other details are observed. See, once you make up your login account, once you actually log into this website, you are allowed to make observations, you are allowed to click pictures, you are allowed to submit those in your repository under a folder name. But that is not required for you to make a project. A project here is a very different thing. You should never open up for a project unless there are enough people collaborating throughout that project, and unless that project is going to be an extensive collection of something. For example, let's say if you want to map all the arachnids in University of Hyderabad, let's say, if you want to say, I am going to do a collection where all the spiders, all the related arachnid individuals, they would all be present in that particular folder. Then yeah, you can make up a project, you can invite people to contribute, because a lot of people would be living nearby and it's convenient for a lot of different people to click pictures. So those people would contribute to your project as well, developing that arachnid whole image data set. And then yeah, it aids in other people's work as well than just your own. Let's try for something else. Can anyone suggest me something? A particular centipede. Yeah, let's try for centipede. Okay, there's one more thing that this platform offers, and I mean, you can obviously, despite images, you can do videos. I mean, you can put a lot of videos, but if you have sound recordings, for example, if you go to some place and you can hear only birds chirping, and you cannot, I mean, see where they are and which birds it would be. You can still record the sound and upload it here in one of your observations for that species. There you would not enter the species name and any other thing. You would rely on a particular, I mean, specialist or your AI algorithm to tell you whether that particular identification was which species and whether you were right or not. So it can also recognize based on sounds of the birds. Hmm. Why is it webp? Yeah. For example, this is another picture of an insect. I mean, I did not. Uh, yeah, here it rightly tells you the I mean genus name. I mean it does not tell you exactly the first suggestion as a species, but in the next suggestion it's exactly that, house centipede. This is what we were looking at. This was one of the pictures that I took from the web, and it rightly classifies it as Scutigera coleoptrata, I think. Yeah, that's the scientific name. So, I mean, this was just an easy work way around as to how the tool works for species identification. And I think this should be it for the demonstration. See, I would tell you all, I mean, I would give you a particular data set. I would transfer to Mr. Srinivas's number, another type of data set, and that would have almost 40-50 images. And based on those images, you can see how the tools work. But because the session was a little, I mean, cut a little short because this was the first session for life sciences, I would also ask you to check for those same images on another app. So please write down the name for another app. It's called Wildbook. Wildbook AI platform. Wildbook. WILD then BOOK. Wild book, yeah. So that works in a similar way, but you will have to navigate through because the time is up anyway. So this that kind of works like this as well, where you would enter, I mean, a photograph or any other kind of identification marker, a sound or a video, and then for you it would recognize the species, and you can also filter through a lot of your collections that you want to go through. A wild book, WILD B. I'll just write up. Yeah. Yeah. No, no, no. See, I mean, most noisy data set, I mean, not even literally and figuratively, would be your noise because it would have a mixture of a lot of things. So, I mean, there are some Fourier transform models you might have learned where frequency of one sound could be distinguished from another sound, even if they are being, I mean, pitched in the same time. So, for example, if a hyena is howling, or if a wolf is, I mean, howling at the same time, you can distinguish the howl from hyena's, I mean, noise. So even if they are occurring at the same point. So what these algorithms would do, they would first try to individualize the voice, first try to, I mean, allocate one particular voice to one particular species, and then they I mean, run the model on that particular frequency for the species. Yeah. Uh, please.
Session material.
Oh, this PPT? Yeah, I'll share the PPT and the data set as well. So I'll just anyway show you how wild book looks in. Wild book. This is anyway the website that you could, I mean, go through later. But all of these platforms could also be looked right up into your phone. Or I mean, if you have Android phone, it would work on Android, and Apple, it would work on an Apple. So there is just no problem at all, and it's more convenient for people to go out and snap a picture from their phone than a laptop, obviously. So it works equally well on that, and you can actually download the app from Play Store or Apple Store if you have an Apple. Uh, the iNaturalist app. Yeah, you can download from Play Store. Yeah. And I think we are done with this session. What is your next session? Yeah, see, I mean, there are different levels to the identification. If a researcher has, yeah, yeah, if a researcher has identified your particular photo as a species, then that would contain more weight. If your algorithm also has identified it as one species, then that would add to that weight. So depending on that, it classifies an image as a research grade or annotation based on a individual, something like that. So it gives you a score. So yeah. Well, anyway, disconnect.
model. Or I can give them a demo in my system. Yeah, that's not a problem.
You want that?
Which one? No, I don't want that. My laptop has a camera. So, so before we begin, I want to give you a brief introduction to the importance of plant disease. So, plant disease causes famine, economic loss. It drives people from one place to another place, right? And also it destroys all the crops and makes farmers poor, right? So it's studying a plant disease is very important. So as a plant disease or plant pathologist, we use all the tools available. We use molecular techniques, we use bioinformatics tools, we use artificial intelligence to detect the presence of plant disease or the plant pathogen in a plant. Okay. So this data I got it from UoH's plant pathology book. So this data is from the US from 2002. We look at it, the attainable crop production, that maximum that we can produce is about 1.5 trillion in that particular year, 2002. But how much the production was there actually? It was only 950 billion, which means we lost around 36% of yield, the production. So the 36% was lost due to several factors. One is plant disease, including bacterial disease, fungal disease, viral disease, and insects. Insects also caused a lot of yield loss. And then weed. So weeds, they compete with the host plant, the crop plant, and then they make the nutrients less available to the crop plant. And that is how also the crop plants cannot produce better grain. So these three factors are more important. One is plant disease and insects and weeds. So they cause almost 36% loss in production. And mainly if you look at this, the losses caused by disease, it costs around 220 billion. See how important it is to study about the plant disease and disease detection. So here it shows that, yeah, so this is the attainable the crop production in trillion, 1.5 trillion, but what they could achieve only is 950 billion, right? So they lost around 36% of production, is almost around 550 trillion, including weed and insect and the disease. Yeah, this diagram also shows similar data where you see the plant disease, it cost more than like almost around 220 billion yield loss due to the disease and crop yield. So what plant pathologists, what we do? So plant pathology is a study of plant diseases, and we attempt to improve the survival of the plants under disease attack. And we also study the causal organisms and the host plant as well as. And then we study how the environmental factors interact with each other to cause disease or to cause resistance. Yeah. So we use all the integrative sciences tools to apply and to cure the plant disease. Yeah. So this figure shows that if you look at the left side, it shows the plants are very important because it fixes the carbon using the photosynthesis, and it fixes gaseous nitrogen to ammonia, and thereby we get the nitrogen from, not only we, all the animals, insects, everyone. So the whole life on the planet depends on plant. Nitrogen, carbon, and transpiration, fruit production. If you look at all these steps can be attacked or prevented by plant disease. If you look at here the root rot. So in my PhD I worked with the dry root rot in chickpea. So basically there's a fungi Macrophyllum officinalis. It's a soil pathogen. It specifically attacks only the root. So once it attacks the root, infects the root, it it kills all the lateral roots, and that way it won't allow the plant take up water. So each disease has a very specific plant stage. So fungus cannot cause disease in all the stages, right? Some causes in the seedling stage, some causes in the vegetative stage, some causes in the reproductive stage. So the disease that I study, it causes only at the reproductive stage. The symptoms will appear only at the flowering stage. So the farmers may think that the plants are growing healthy, right? But when it reaches the flowering and the early seeding stage, you will see all the plants become yellow color, straw yellow color. So that leads to a lot of chickpea yield loss. So one is root rot, the crown gall, it's very famous, we know. So not only they kill the roots, the leaves, it also makes cells divide more. So in case of ground gall, there's uncontrolled division of cells. It's called hyperplasia, and also it makes the cell size bigger, right? So it's called hypertrophy. So in case of ground gall, you can observe these two phenomena. One is hyperplasia and hypertrophy. More number of cells and bigger number of cells. Yes, that is. And vascular wilt, you might have heard fusarium wilt that specifically attacks vascular bundle. So if a pathogen attacks a vascular bundle, what it can do is it can block the movement of water from root to shoot, right? So if the fungus block the water movement, then the plant cannot take water, and then it will wilt, and dry. And so these are the wilt symptoms, so that they droop or they dry immediately with the yellow symptoms in straw color. And there are cankers, there are leaf spots. The leaf spots play a very big role because leaf spots, the leaf area has to be maintained green color so that the plant can fix more number of carbon using photosynthesis. If the cells are dead and if they see a leaf spot, that means the plant cannot fix with maximum carbon using photosynthesis. And obviously the fruits. So when the farmers get into a fruiting stage, at that time they get infection, fruit rot, fruit spot. So these are very economically important diseases and plant parts that we study and we try to cure them. I will give you in the next slides, upcoming slides. So the root rot in chickpea is Macrophomina phaseolina. Macrophomina phaseolina. I can write it here. No. So I did my PhD from NIPGR. It's in Delhi. But I used to visit Yogi Vemana University for a field trial. Is there anybody from Yogi Vemana University? No? Kadapa. Okay. So we have a collaborator, Chandra Obal Reddy sir. Yes, so he is an active collaborator even now. So I used to go visit farmers' field and collect samples. Then we published a lot of papers with him. Macrophomina and phaseolina. Yeah, this causes. So the chickpea has been faced with a lot of pathogens, not only this Macrophomina, also Fusarium oxysporum and Ascocata arabiae, which causes Ascocata blight. But all these diseases need certain environment, certain favorable condition. So Macrophomina, it causes dry root rot. So the plant has to be under drought stress, then only it can show the symptom. But in case of Ascocata arabiae, which is another disease which causes Ascocata blight. This one. So this disease is uncommon in southern part of India, especially in Andhra Pradesh, because we have little hot weather. But this is more common in northern area where they get winter. At the time we see a lot of disease incidents. This Ascocata blight. Yeah, so that also play a role. So when we predict disease occurrence, we need to not only consider the fungal pathogen presence, but also the weather condition, whether it's a dry condition or cold condition or humid condition. All those has to be. So it's not easy to predict the occurrence of, unless we add all the factors. So these are the basic the pathogens that can be a potential causal agent: fungi, bacteria, molecules, parasitic virus, and nematodes. So nematodes, they cause root knots, right? So just for your awareness. These are some basic, very commonly occurring, economically important disease. So the moment I say economically important, means they really cause economically ill loss. Okay, they even drive people from one place to another place. They even collapse economy of the country. Yeah. So I hope you all know the Irish famine. You know Irish famine. There was a famine in Ireland. It was due to a phytophthora blight. Yeah. So even we had a Bengal famine, right? So it was due to a weed disease. Yes. Yeah. Okay, so now we have little like introduction to a plant disease and its importance, right? So one of very important thing that we can do to prevent this disease progression or yield loss is to detecting the disease at early stage. Okay, so for that purpose, we have two models here that we can train by ourselves, then we can see. Because I am also from biology background, so I'm not systems biology where and I don't know how to code and all. But what I can do is there are two components in case of predicting or detecting disease. One is knowledge about the plant pathology, complete pathogen, complete its epidemiology, and complete its life cycle and disease symptoms, developmental symptoms. That is one part. Another part is on collecting the images. Another part is we take the expert from systems biology department and we ask them to write a code using C++ or Java or other models. Okay. So we play a very main role in collecting the images and setting up all the stages and classifying them so that others can use the data and develop a model. So one of them is in our today's talk. One is teachable machine. It's a freely available model. It was developed by Google. It's very simple. So basically we can train the model by giving input, different types of images, like for example, healthy plant images and diseased images, right? Likewise, we can input and then we can ask that particular model to predict which one is healthy, which one is symptomed, right? So we'll do this one for now with our own. The first one is where to gather the data, where the biologists play the bigger role, that we gather the data and capture the images and different stages. We will see one of the YouTubes.
Hi, I'm going to show you how to train a model with teachable machine. Like all machine learning workflows, you first need to gather data. What you see here on the left are what we call classes.
Okay.
Each of these represents a different category you want to teach the computer to recognize. So let's try adding data to our first class. I'm going to call this one neutral. You have two options here. You can upload files from your local computer or use a webcam. Let's use a webcam. Hit record and it can start collecting images. You can see that I'm moving around here to add variety to my data set. Let's try adding data to our second class. I'm going to call this one dog. Again, I'm going to hit webcam and try to get data from the stuffed animal. You can again see that I'm moving it around to collect some variety. All right, now we have enough data to train this model. A teachable machine doesn't just work with image data. You can also train an audio model, and the interface looks pretty much the same. One difference is that the data is collected in the form of a spectrogram, which is simply a different way of visualizing audio. Another difference is that you need to record one really long background class, 20 seconds of it to be precise. And finally, all this audio data gets extracted into 1 second samples. Like, hi,
Yeah.
I'm going to show you how to train.
Basically, this video will show you how the Google Teachable
In a model of
model will
machine.
work.
Like all machine learning workflows, you first need to gather data. What you see here on the left are what we call classes. Each of these represents a different category you want to teach the computer to recognize. So let's try adding data to our first class. I'm going to call this one neutral. You have two options here. You can upload files from your local computer or use a webcam. Let's use a webcam. Hit record, and this can start collecting images. You can see that I'm moving around here to add variety to my data set. Let's try adding data to our second class. I'm going to call this one dog. Again, I'm going to hit webcam and try to get data from this stuffed animal. You can again see that I'm moving it around to collect some variety. All right, now we have enough data to train this model. A teachable machine doesn't just work with image data. You can also train an audio model, and the interface looks pretty much the same. One difference is that the data is collected in the form of a spectrogram, which is simply a different way of visualizing audio. Another difference is that you need to record one really long background class, 20 seconds of it to be precise. And finally, all this audio data gets extracted into 1 second samples, like this. So you can make a recognizer that recognizes something short, like clapping or whistling. Great, now we're ready to train this audio model. And finally, you can use Teachable Machine to create a pose model. Pose estimation is simply a technique for tracking key points on your body, so that you can train the model to say, recognize when your head is tilted left, or when your arm is raised. All right, now we have enough data for this model too. One thing to note is that the entire time you're capturing this data, it's staying on device. It's not being sent to any server. So if you want to keep your data, you can either download your samples or save them to Drive. Look out for the next video on how to continue training your model.
Okay, so I'm going to train myself. So basically, like we may wonder how the model differentiates between two different samples, between healthy or diseased ones, right? So they have created a model from the Google platform. So where if I capture my images and source image, and we train the model will be able to differentiate between me and sir. For example, like let's go here and get started. I'm going to go with the image project, and we'll go with the standard image model. And I'm going to say Vadivel. And I'm going to feed my image into the teachable machine. Yes. Can I have volunteer here? Where I need their face. Any volunteer youngsters or sir, please come. Sir, you write your name, sir, and then. Yes, sir. Then you can take your images, sir. Hold, sir. Hold that. Yeah. Yes, sir. Thank you, sir. Sir, please stay for a minute, sir. Sir, Please stay for a minute. We have to show that. So now we have given, please. Now we have given our input, two different images. Now we are going to train the model. See, all the AI based or machine learning based or computer vision based models, they follow the same pipeline. Okay, so we have to input our images, then we have to train that model, particular model, to differentiate between two different images or two different objects or whatever, or two different sounds, right, or postures. Then it will train itself. Then it will be able to differentiate two different people. So now we are training it. It's going to take some time. During the training, we should not close this tab, particular tab, otherwise it will get disturbed. Okay, so now it has trained. You see here, Vadivel and Srinivas. So if I go here, see it's saying that Vadivel is 100%, right? If sir comes, can you please show me? You may have to sit there. You see, it has detected. Yes. So this is how we can train this model to differentiate between two different people. We can also yeah. So do you have webcam in the system? No. What? You can upload. If you have, so I think you have an image from that website, right? That database. Yes. So let me do something for you guys. So go to new project, standard image. Do you have access to these files? This potato light and all? Do you have? Yeah. So there is a website. Let me help you out. So you can get the image from this website, Plant Village dataset. Please go to this website, Plant Village dataset, and you can download it from this website. You will get all the images, the healthy one. Yes, sir. Yeah, here you can download. Oh yeah, only the data set. Oh, you can sign in. Let me ask you to sign in, but yeah, please go ahead and sign in. Ma'am. Ma'am, at least you need 20, ma'am. Yes, ma'am. So in order to train your model, you have to give more number of images. The more number of images that you feed into the model, the accuracy will increase. The better the accuracy. Yes, ma'am. Plant village data set. Yeah, the data set. Any data set. Yeah. You can download from here. And download that file. Yeah. Yeah, from anywhere. No, can you go back? Can you type here only the plant village dataset here? Just type again, like it came differently for me. Go to the first page. It's download this one. No, this one, this download this one. Yes, and this one, this data set. Yes. Yeah, see it's downloading here. Yes, ma'am. Yes. Not the code. If you know Java coding, you can download and then you can play with that. Yeah, this one. Oops, FDP. You can register with just sign in with your Google. Yes, sir. Ma'am.
You have to download these DCFs.
This one.
Then download
And this one.
as zip.
Yes, ma'am. Ma'am, you have to sign in with your Google, or you can register yourself. Yes, ma'am. Email. Yes, sir. That one.
Is to be downloaded.
All will be downloaded. This you can download and then this zip file. Yeah. You may have to sign in with your email.
Correct mail, but I am getting as incorrect password. Different password.
Okay. Can you go back, ma'am?
Huh?
Can you go back, ma'am?
Back up.
Let me help you, ma'am. Let me help you.
Sign in with the Google.
Which
Oh,
is
that's one. That's one.
Ma'am, it will be in JPEG form, ma'am. You can download here.
Zip form.
Click this one. It will download on itself. Yeah, you have to sign in with Google. Oh, you already have an account, ma'am. Oh, that's great. We'll sign in, ma'am. You can sign in. Password is already there, I guess. You can sign in here.
Okay, Kagal.
Oh, you have access to this, Raka. Oh, that's great. Now you can download here. In this. Yes. So you can use any number of data sets, not only this. Yes, ma'am.
Username is already taken.
Yes, you can change, ma'am.
All the three, I haven't recognized in orange.
which means you have to give more number of images. How many images are there? Orange. You have to give more, like at least 20, 20, 20. You can give more.
We have to upload in this.
Yes, you upload. So please write here. It is Apple. The class one you can edit here. No. Let me help you with that. So this one, if you go here, you change this list. So you can select apple from top to bottom and banana top to bottom. Okay. Yes, sir. So here you can down. It's downloading. Yes. Sir. It's at this one only. This zip file. Zip. Yes, sir. Downloading. So I hope you got. Okay, ma'am. Wait 1 minute, ma'am. I'm coming. Yes, ma'am. No, ma'am. It's not downloaded yet. So download here. Go here and here.
Sir, you want show on the
Okay.
Twitter. It will be easy for everyone.
Okay.
The Gmail login.
Okay. I think there's a issue. Let me show you on my display. So this website is a reliable website, so you don't have to worry about your data compromise or anything. So Plan Village dataset. Go here. Actually, already I registered myself. Okay, I made an account in their website, so I could download it directly. Yeah, so it will download directly. If you don't have a website, if you haven't registered yet, please register. It's a very good one. So you can use the same dataset to train your students in your colleges. I hope everybody has the data now. So let's go to so if the data set is larger, like more than 100, 200, it will take some time to train the model. So I have started training maybe 10, 15 minutes ago, and it's still training. So now it is done. So now okay, so now I want to so I made two classes. One is plant potato healthy and blight disease. Now I have trained the model. So suppose if you are growing a potato cultivating in your farm, you go to the farm, click a picture and upload into this tab. Okay, so it will tell you whether it is healthy or blight infected. Okay, so I'm going to upload two images. So first I'm going to give blight. You see, the moment I uploaded the data, the file, it says blight, disease 100%. There are several types of diseases, symptoms. We will upload all of them, and then we will see if it is giving us right data. This is completely a healthy image, so it shows 100% potato healthy. Can you do? Is it working for you? No. Okay. Meanwhile, what I will do, I will train all the data. I'll put all the data and then train. So we can give any number of. Let me check what is there in my table. See, one file has 3,208 sample images. So I have given input healthy potato leaves, blight disease, yellow leaf curl, and bacterial spot. Now my model is getting trained. Let me help you to load your data into the model. Shall I help you, sir?
Can I load the images directly to the models? So actually, the model.
Sir, you have downloaded here, sir. Let me check, help you, sir. Here it is. Let's go here and open the zip, unzip. Yeah, extract. Extract the images. Then from here we can upload the images.
Okay, sir, like we have to open a teachable website after. Like after
Yeah. Yes. So once you download all the data set, then open that teachable website and go to image project and the standard image model. Yes. Now you start with the class 1. So the class 1 you have to change the name. So basically you need to know what data set that you have. So I had healthy and disease symptoms. So you can first go with the healthy one. Healthy leaves. Then class 2, you can go with any disease. Yes, sir, it may take some time, sir. It's extracting the data. So let's extract the data, sir. So I think, so you have an archive, right? You have to unzip the archive. Extract all the images to a different file.
How to give?
Yeah. So you give brinjal white and brinjal red. Just train, ma'am. If you click train, you train. Click it. It is training now. Preparing your training data, it will take some time. Okay. So don't close this data. It's already trained. How many images were there?
We have 20 images.
Okay. That's cool then. So turn on your input. It's on. So go to file and then select any one of the images from your data set.
No, downloads down.
Download.
SM file. So, but first one.
Yeah, it's brinjal white. It says brinjal white is 99% correct, and brinjal red is 1% correct.
This one or this one?
Yes, this image.
How can it be? So we have target brinjal white for this. Brinjal I think we
Is it brinjal red?
okay. Then it is correct.
Yes, ma'am.
Maybe fruit it is confused.
So in order
These are the label. They have, I have to unzip them because they are zipped
please
file.
unzip, ma'am.
How can I unzip?
Ma'am, okay, let me do that now.
Because it is not taking the images.
Stuck here. Okay, let's. Okay. This one, ma'am.
Extract all.
Its folder empty. It says.
That one, first one. You do.
Actually, it says empty, ma'am. Label my project. Only 3 KB it says, ma'am. 3 KB is very less. Okay, so let's download again, ma'am, if possible.
I'm not getting downloaded.
It's downloading.
Download. Download.
It's downloading, ma'am.
Okay, okay.
Yeah, it's going to take some time.
Okay, wait.
Yeah. Yes. So, yes, ma'am. So, in order to improve your accuracy, you have to input more number of images. Tens and thousands of images you have to give. Then you will get accurate prediction. Okay, sir. Here, then we'll go. Sir, before that, we into label. What do you want to input? Like healthy. You can give. Yes, sir. So now we'll go here, and so we have a healthy leaf here. Let's go with potato. So we have potato, tomato, and everything. So we'll go here. So now we'll just press Control A. All the images that we are selected, and we are loading into the sample. So like this, you can add another disease, sir. So you go back here and see what disease that you want to take. There are several diseases in potato, 1, 2, 3, 4. You can select whichever you want and then put the names, and then you can add all the images. Yes, sir, you can add more, sir. Open it now. Yes, what happened?
Here, ma'am is unable to add the file.
Okay, so I think it is noise. So go back here. Yeah, so we can give in 3 things. One is image, audio, and post. So you better select this one. So you can select this one, standard image model. Now you can upload your all the images. So yeah, plant village. So before you do that,
It is not coming now.
it's not there. Okay, let me check. Hey, it's here.
Yeah, got it.
So before you, so this one, which one? This one, okay. So before you, okay. Yeah, go like this. So yeah, so she has given name, right? Plant healthy, rotted. Likewise, you have to give the name here. Whatever image that you are going to upload, give the name, change the name, upload your images, and you can add more. Yes. Yes, ma'am.
Here we did.
Great. So you trained? Great. Yeah, absolutely. So whatever image that you upload, it will tell you whether it is healthy or symptom. This is infected. So input is on. Can you go here, webcam, go to file. Yeah, now you can upload. Ma'am, can you hit the webcam? Hit the webcam. Okay, go to file. Upload the file. Right? Yeah. So, so we don't have any sample data, right? So we are training the model with the number of images, and we are uploading the same image to, we are asking the model to detect what is it. So obviously it will detect 100%, right? So we should have some kind of sample images. Maybe you can try at your school or home. You can go to any form, click images, you can upload them, and ask the model to predict whether what kind of disease it is. So if you are done with the one disease, please add all the diseases. So you have potato, tomato, and pepper. Please add all the classes and try if that model could predict different plants and different diseases. Okay? So I'll give you one. There is one app. This app is very famous. So one minute, sir. Are you aware of this app? This Merlin Bird ID. This app was developed by Cornell University, which is a very famous university in the US. So if you have a bird in your home, turn on the app and record its voice. It will tell you what the bird is. Okay. So they use similar model that what we are using. Okay. So right now we are uploading the images. There are two more categories. One is you can upload the sound, train the model, and then it will predict what kind of. So while developing this app, they might have uploaded all the unique sounds of different birds, right? Like us, like in hundreds numbers. They might have trained the app. Then whenever we record the sound of that bird that you encounter, it will tell you what the bird is. So next we will see one of the apps that we can use to detect disease. Once you complete this session, right? Can you upload all the images, different diseases, and then see if it can train and predict, right? Then you not one by one you are doing. Okay. So 173, 108 number.
Yeah.
Oh, that's cool.
The number should be.
No, the more the number, better the accuracy. You add if. Okay. Is it predicting nicely? Yes. Obviously, it will predict because we use the same images. But if you get an image from your farm, it might give you maybe 70% or 80%. Yeah, does it? You should detect. It is not tomato. First of all, the plant is different plant. Yes. Yeah. Yes. Is it correct?
Yes, sir.
Okay. Okay, yeah. Yes, sir.
I tried 4 types of classes.
Sir, you have to add more number of.
How do you write the identify this is of the plant?
Okay, so here you go to file. Now you can upload
the file?
Yeah,
Otherwise, okay.
you upload this image. You say it's leaf light, right?
Okay.
It says put it up light. Like
It was gone. I think.
yeah, so it shows like so you need to train more. You give only 4 samples.
Yes, sir.
No, no. Actually, wait. This is not con.
Okay.
So you have to give same images. So it will train. So if you are giving con, give con all the images. Then that accuracy will increase. Right? If you give it tomato, only give tomato. Yeah, you can give more images. We add all the images, like 3000 images. Upload them into. So archive the file, sir. Not that one. That's a zip file. Yeah, can you open that one? And plan village again. You can double click there itself. Double click, sir. Yes. So now we can give healthy one. Let's give healthy potato. So how many images we have totally? I think there will be 3000. So can you press Control A? Control A. Yeah, you select all the images now. It's uploading. Totally 152. You please change the name of this one. So this is healthy potato. So did you download, sir? And this one, zip file. Yeah, it's downloading now. It will take some time, sir. It will take maybe 2-3 minutes. So likewise, you can add images, different categories. I'm going to change the name. When you add 2 different classes or 3 different classes, then you train the model. Go here. Yeah, that one, and go back here. Let me help you. So go back here. Suppose if you are interested in
Potato leaf.
potato leaf light. Okay. So Control A, please. Let's select all of them. Yeah. Then yeah, it's going to upload. So plant village. Double click. So double click. Now open this. Yeah. So here you have all the categories with all the images. So you can first go with the healthy one. This healthy. Or you can go with the pepper healthy. Yeah, and Control A. You select all. Then can you click? Okay, got. So 1 minute, sir. Yeah. So here. Okay, I'm sorry. It's going to open all the images now. Sir, I opened all the images mistakenly. It's 3,000 images. No cost. So actually, no problem, sir. Yeah, so here we will upload your images. So for example, you had download and archive. Okay, I don't know where that file is now. So let's find that file. So let us complete, sir. It's going to open. There are 3,000 images. Model train, sir. Okay, so now we can do one thing. So go up. So this is a file.
Yes.
So now you select the image that you want to identify. Suppose you can open any one of the image and open. Okay, so yeah, it says there class 2. That's why you need to change the name of this one.
Blight.
Yeah, you can name it. Change. Yeah, you can write the blight. So let me open all the images. Then we'll close. Yeah, there will be a lot of images. We can keep changing the feed, sir. We can feed more images here, over here. Like you can drop different kind of images and then ask the tool to predict. So how's the prediction? Is it going fine? Yeah, it says healthy, but it's not healthy. So you have to train more number of images. So you are giving only three only, right? So this also. So more image that you input, better the accuracy. Okay, give 3,000, 4,000 images. If it is there in the file, upload all of them. Train the model.
Could upload here.
No problem, ma'am. Here, ma'am. Okay, so can we? This one, this nothing, ma'am. This just to scroll, not a problem. Okay, why it is there? Maybe get it strong.
Times I did, I got it, but now.
Okay, maybe can we?
When we are doing in a laptop, when we have a webcam, then we can directly show the leaf.
Yeah. Then can you train again? Maybe it says some glitch. Okay.
It needs more amount of data.
It needs more data. So another thing is with a model. Yeah, the model still needs improvement. 10,000, 20,000, even 1 lakh images. Okay. Yeah, please train more. So whatever we are doing right now is a very basic training like session. I will give you few more examples. Yes, sir. Yes, we got it here. So just upload any one of this. It will tell you. So this says it's a spot. Is it correct? It says spot is 100%. It's not healthy. It's not early blight. Only spot. Go here. Maybe you can take another something else. Is it all tomato, sir?
Tomatoes.
Maybe you can take this one of this and then select. It says 67% early blight and also spot. So you can download this in your app, mobile, sir. Yes, sir. That app is really nice. This one. If you visit any bird sanctuary, you can identify the bird with this one. Yes. So I hope you all got it. So should we move to next level? Yes, ma'am. Okay.
But here I put the last thing.
Minimum you have to like take at least 20 images. Minimum 20 images and maximum as much as you want.
Bird image in the class 2.
Okay.
But the system telling that 50% this, 44% this.
So that model algorithm will detect based on size, shape, everything. So if there is any shape similarity between these two, it will going to give you 50% or 20%. Yes. So, but if you train more images.
Diversity.
Oh, that's great.
Fish one and diversity.
Okay.
That's why I want to check with fish.
Ma'am, same fish, different angle, you upload 1,000 image. Okay. Then you see, it will give you accurate prediction. Single image. Ma'am, you see one person today. After 1 week, they ask you, okay, identify that person in among 100 people.
Information about the single fish. Then how can I?
So single fish, different angle. Take 100 images and upload. And then train the data. It will give you accurate. Yes. So more number of images, better accuracy. That was the thing, right? So I hope all of you have got the tool how to use, right? So this is a very basic version. So now, suppose if you are, if you know suppose if you know Java or C++, we can do one thing. We can go here and alter this codes to improve the accuracy. Okay. I don't know anything about the code. Okay. I take the help of my collaborators. But in case if you know, or your students or your son, daughter, if they know, you can ask them to look at the code and edit the code to improve the accuracy that you can do. Okay, so you can download this code from this website. Whatever you have done, it is there, or you can. So for right now, all these images are not stored in the website. Okay, so it will be never stored. So, but if you want it to be stored, please export the model, and you can download the data. You can go here on the left side. You can download here. Download project as file. It will have all the codes and all the images that you have submitted to the model, and all the steps how you did. Everything will be there. So now I'm going to see if the model can differentiate between this blue cap and black cap. Okay, with me in my image. So in order to improve the accuracy for the minute details, we need to again give more number of images in different angle, different places, so that it can recognize. So yes, this is done. Yes. Now I'm training the model because so as I mentioned before, it's very important to detect the plant disease at the early stage so that we can apply fungicide or any other chemical agent to control, or we can uproot the particular plant in case of viral infected, and we can burn them. If we don't detect it at the early stage, then the later stage, the advanced stage, it won't be easy to cure that particular disease. Okay. So early disease, the symptoms you may see here: potato blight, highly like burnt one. But in the early stage, you will see only the small speck. It will start with small speck, then slowly it will become bigger in area. So if we can train this model with early stage images and detect it, just remove the plant, or you can cut that particular branch, or you apply fungicide. Right? So now let's see if it can detect. So I have two kinds of classes, me with a blue cap and black cap. So it's already saying blue cap. I don't know where it sees a blue. Maybe it sees a blue somewhere behind, right? You see how accurate it is? Yes. Because I have given 400 images. If we given more images, like 1000 images, then it's going to be really accurate. Right? Ma'am? What is that, ma'am? Yes, ma'am.
My file.
Yeah, that is your file. So the codes will be in Word, Notepad only, ma'am. Yes, or in because yeah, this is just once. No, you have to open. So the file format will be very different in case of this one. TM file.
TM file it is saying.
Maybe. This one, ma'am. Where is the file, that website?
Which way?
That,
Downloaded file?
no, the where do you want to download?
Download.
This one, ma'am, yes. So go here and go my project file. What TM?
Project team.
TM is training model, ma'am, but I don't know exactly where to. This can be opened in maybe in Java or C++ only. In that, it can be given as an input file. Yeah, we cannot open in the Word file or Notepad file to work. No. Or if you have a this terminal file, then where they do a coding, in that it can open.
Not file.
No. So the project file that you download, that you can take the file into either C++ Java page and then you can paste them. It cannot open your Notepad with the same codes. So that is there.
Final output.
So you trained a healthy one.
Yes.
And bacterial spot.
Okay, sir.
Suppose you have some image from your farm, bring that image and upload it and then see what kind of. So this is healthy one.
Okay.
So it is showing healthy 100%.
100%.
Right? So let's upload
Unhealthy.
unhealthy one. So maybe bacterial spot. Just choose any one of them. So it shows 100% of bacterial spot, which means that so right now we use the same images to train the model, right?
Okay.
If you have some unknown leaf, unknown one, you upload it into the into this site. It will tell you whether it has an infection
Or not.
or not. Yeah, just to identify the disease. Yes. Okay, sir. Okay. Now I'll help you. All open, no? Let's close all this. Got hanged, it seems. Yeah, it's coming up. Yes, just disease identification.
Sir, in the student point of view,
Sir, actually, it's not that simple. There are several diseases, right? So, if you feed, like you have only fed only spot. If you feed viral images or bacterial, fungal images, so it will tell you different type of diseases. Here. Yes. So here you have only two classes, right? If you add few more different type of disease symptoms, then it will tell you.
Suppose we don't.
That's the first point. So normally we have already all the images of catalog. Like a plant can have only this many potential diseases. Maybe 5 diseases, like in case of rice, it can be rice blight, right? Something like that only we can take, and then we can train the model. Suppose you go to a field and you don't know what is the disease is, get the image and then upload, it will tell you. Yes, yeah, the model will identify. Okay. So next, after lunch, we will look at that app. There is one app, Plantix. Yes, that app you can download in your mobile phone. You can install. After lunch, we will see how to use that app. Let me show you. Ma'am, you can improve the model, ma'am. The script you can improve. The algorithm, you can improve the algorithm into a better version. Yes. So the same model, you can make this model into an app by yourself. So how the app's running in our mobile phone? The same codes only. Yes, all the WhatsApp, Facebook, everything is running based on a code. Yes. Yes, into a better version of this one, into a separate app. You can improve the accuracy. You can change the parameters. We can add number of classes. I don't know how many classes are that we can upload here. We can upload like maybe 100 classes. You can change the model. Yes. So, is it working? What happened to the projector? Yeah. So we gather the data from the data set, right? And we train the model. No, there are 2 ways, man. One is webcam for your own simple training, but the main purpose is to feed the data from the plants. Okay, that's the purpose. Webcam is only to show how that model works by a trainer. So, but the important thing is to load your plant data and then predict the disease. Then we can export the file and then for further improvement of your program. So this we will see after lunch, the Plantix. This is the app. So unfortunately this app is not available on iPhone. If you have iPhone, I don't think you can install this app. But Android can install. Right? Yeah, please go ahead and install and we'll see how. So now with the teachable model, you have to bring the image to the laptop and then feed and then detect, right? But with this app, you can directly go to field, take an image and upload into the app. That app will tell you what disease it is, right? Yeah. Yes, please come. We were almost done. Yes.
You have to complete your lunch by 2 o'clock. The class will start at 2 and ends at 2:50. And after 2:50, you have other classes. You have to complete the lunch by 2 o'clock. You have to start at 10. Class is that you have lunch in room number 5. You complete your lunch at 2 o'clock. You have to come here. The class starts at 2 o'clock again. Ends at 2:50.
Okay. So we will do 2 things. One is the Plantix And there's one more app. We can predict the yield, how much yield that we can get. Okay, that's another app that we will see how to use that model. Ma'am, I'll tell you, ma'am. Yeah, I will let you know in the class. Okay, yes. Okay, then let's go for lunch. Then we'll come back by 2 o'clock. Then we can start that. I hope I am okay to explain, right? Yes. Was I clear? Okay, let's go. Thank you so much. So we have very less time, so we will start right away. So far we have seen the teachable model, right? So now we know if we have a set of already known images, right, we can use that images to train the model, and then you can predict whether diseases there are yes or absent with a new image, right? So now we'll use this app, Plantix app. So I request all of you to download and install the app in your mobile phone. Yes, mobile phone. So it's not compatible with iPhone, but with Android. So these are free apps. We'll watch some YouTube. So basically this is the app, Plantix. So very similar to the previous topic. So this app also has a library where they have so many diseases by insect, by fungal pathogen, viral pathogen. So there are 3 columns they have here. So you can go to the crop. I think someone is very interested in brinjal, ma'am. So you can select the brinjal, and then you can select the pathogen, virus. So it will give you only the virus caused disease in brinjal, right? So scotty mold, black shark, wet rot, right? So this app has a library of ton number of images from different diseases for different crops, right? Here, this app is already trained, which you have just done using the teachable model, but this app was already developed. It has all the trained images. Now, you take the app, install it, and then take a picture of any plant that you want to detect, if there's a disease or not. It will tell you what disease it is, what is the pathogen. So let me give you a small, yes. So there are 4 components for plant pathologist, right? So only the disease, what disease it is, the agent that caused the disease, the causal agent, right? The disease can be any disease, bacterial, spot, or cold. And the agent is maybe a bacteria, fungi, or virus, and the disease symptoms, right? So in case of tomato, there are so many diseases. The first one is most important is mosaic virus. It is caused by tomato mosaic virus. The symptoms look like this: some kind of cold and yellowish. There are a few more diseases that I'll show you. Yellow leaf coil virus. It is caused by another virus. See, the viruses are unlike bacteria and fungi. They produce spore, and the air carries those spores, and then like transfer the spores and then transport them and then land the spores on newly uninfected plants, right? But in case of virus, it doesn't produce any spore, right? But this whitefly, they carry the vector. They are the vectors. They carry the virus. So when they feed on a particular plant sap, it will take some virus, then it will go and feed on another plant, and then it will deliver the same viral particles, right? So here I'm not showing the fourth component. That's very important component that we will discuss tomorrow. So if you know disease, if you know the causal agent and if you know this symptom, it is very easy to control the disease, right? The control, I'm not adding it here. So tomorrow we will see the theory. What are the agents that we can use to control a particular disease, particular crop. Okay. So in our model, previous model, so we had the crop and we had the disease, right? So we trained the model. The current app has all the library already trained. So just install the app and take the image of any plant. It's supposed to show you what is the plant and what is the disease and what caused the disease, the causal agent. Okay. So unfortunately, I couldn't download that app. Were there any volunteers with the Android phone? So I can see how that app looks. Yes. That's great. Okay.
Select crops. So I have selected 8 crops.
Okay.
If I click on that, it has came here in this window.
Yes.
So click on the picture of the particular plant.
Yes. Yes.
Diagram.
So there are 3 options here. One is take picture, see the diagnosis, and get medicine. Right. So the medicine is nothing but sorry. Yeah, you can go ahead. We can maybe make the image bigger. Yes, ma'am. The whole frame should be plant. Okay, there shouldn't be any black. Maybe we can do one thing. We can maybe you have downloaded the previous session, and that you can open them. Okay. We can go closer a little. Yeah. Let's try this. Of course, it will take right. So you can. So you have downloaded some images. Same image. So you have the image from the previous session. Upload in progress. You're on to the point, sir. Thank you. What is the crop, sir? Check here. I don't know what crop is it. I don't know. Quite different. You can go closer to it. when you capture the image, the camera frame tells you if it's going to be good picture or you have to move closer or further, right? So it will, yeah.
Maybe it will be more.
Yeah. So these are all algorithms based model. The author who has trained this model might have trained this one with a lot of pictures, different crops, different diseases. Right? So with the current government, so basically we do basic science, right? Basic science is not enough because basic science doesn't reach the public, right? Who is giving us money to do the research. So now the government is asking, adding a component in the project that whatever we do, it should reach the public in the form of application or app or any tool. So that is another purpose that drives this program with a yeah, I just want to give you some. So in our lab, I mentioned in the beginning that I worked with chickpea and dry root rot pathogen, right? Disease caused by Macropomena fasciolina. So this is our paper from my PhD we published. Yes. So this Rajendra Sinha is my senior and on myself and our collaborator from UoH University, Chandrabal Reddy, and there are other people. And this is my supervisor, Tapas Andul Kumar, TNIBGI. So what we did is, so a pathogen alone cannot cause disease. It needs favorable condition, right? If there's only pathogen and the condition is not favorable, it can cause disease and it may cause maybe 10% yield loss or 5% yield loss. It cannot go beyond that. But the environment is favorable. Like for example, in my pathogen, there should be no water. Like if the crop has no water at the time of developmental stage, and then there's a pathogen infection, that will aggravate the disease incidence and severity. Right? So we not only used the pathogen, but also we used another factor, soil moisture. Right? There are two different diseases in case of our study we observed. One is dry root rot with less moisture, no water. Another one is Ascocota rabi, for which there should be a lot of water in the field and it should be really humid, 100% humid, and a lot of winter should be there. Then only the pathogen can germinate on the plant leaf and cause disease. Right? Yeah. Just to you see here, so we have the dry root dot here, on the y-axis we have temperature and we have rainfall. So temperature and rainfall, they play a very big role because they decide whether there will be a drought or not. Right? So in case of Andhra Pradesh, we have high temperature and low moisture. So whenever like right now how we did, so if you look at the C1, there are some actual disease incidents, right? That is our data. So we train the model with the known data. So every year we go to field and record data, record soil moisture, and then disease incidents percentage. So we have data like 10% soil moisture and 90% disease incidents. Like that we had a data. So first we trained the entire the model, the artificial neural network. Then you see the I think the red one is our actual one, the green one is the predicted one. Right? So we gave only one data. we gave only soil moisture data. If you have soil moisture data, what could be the prediction? So it predicted that more disease incidence and more disease index. So it predicted in a favorable manner. So whenever there is a low moisture, there's high chance of disease severity of dry root rot. Yeah, that is our study. That whenever we propose our result, so not only we do only one experiment or one statistics, we do several statistics. So with 4 or 5 statistics tool, we prove. So this is one of them. That whenever there is a increased dry root rot, we see increased amount of potassium in the soil. So there is a correlation between these two. So this was published in 2020, I guess. Yeah, 2021. Yeah. So now my juniors, they are looking at how these different components, they interact each other, and they are trying to understand now in the lab. Yeah, these are the disease incidents that we scored in the field. Yeah, and also we study the root cross section. So you see that there's a control one, there's an infected one. So in case of crop, there will be always a resistant crop and susceptible, sensitive one, right? So we used this 4 and 5 with this resistant one, and this one is a susceptible one. See, these are really highly infected compared to the resistant one. Yeah. So I just want to share my paper with you. Nothing much. So could you download the app? All of you? Okay, then we'll move to the next topic, which is yeah. Can you please download this software? We already have this one. That's great. So do you know how to use this?
Some part.
Okay, then it's pretty much cover the same thing. Yes, so we will have the disease incidents and all the data. We will upload into this tool. Then it will give us graphs. Right? Validation means, then how? Then this will give you like the graph I made, right? So it will give you all the graphs so that you can correlate the actual versus predicted data. So yeah, it will just give you visualized form of your data. Yeah. So then in that case,
with icons of oranges components on the left.
Ma'am, did you watch this video yesterday? Oh, let's run this video once. So there are several statistics tools like this. So it's just one of them. So in research we use Prism graph pad for our analysis. I think this one is works well with the AA, I guess.
Assuming you have already downloaded and installed Orange on your computer, we can begin. Orange opens with a welcome screen. We won't need it here, so I'm closing it, exposing the main window of Orange. There is a toolbox with icons of oranges, components on the left, and the blank canvas on the right. Orange supports the analysis of data through the construction of workflows. Orange workflows contain components that read, process, and visualize data and models. We refer to these components as widgets. Let us start with the datasets widget, that can read data that has already been prepared on the Orange server. We place an instance of the datasets widget on the canvas by clicking on its icon. Here it is. Now I will double-click on the icon to expose the content of the widget. It lists various datasets, and I will use the one on socioeconomic data. The dataset is called HDI, and let me find it by typing its name in the filter box. Here it is. To load the data, I will double-click on the HDI row. Notice a green circle that indicates that the data is loaded. The widget reports 188 rows and 52 columns in this dataset. But where is this data? The dataset widget just loads the data. To display it, we need another widget called Data Table. I will place it on the canvas by clicking on its icon. If I open the Data Table widget, I find it empty. This is because the data we loaded with the Datasets widget has yet to be communicated to the data table. I will drag a link from Datasets to the Data Table widget to display this communication. The Data Table widget displays the data as soon as I do this. Orange widgets communicate. As soon as you change something in one widget, it outputs the results, and the widgets downstream in the workflow update their content. For example, I can open a datasets widget and choose some other dataset, say on liver disorders. Notice the immediate change in the data table? Let's switch back to the HDI dataset. Here it is. It contains the data on 52 countries that are described with 52 features. The first two features are the country's name and Human Development Index, and all other features describe some socioeconomic features. For instance, the first one provides average life expectancy, which is the country where they live longest. I can click on the column heading for sorting. It seems you would need to move to Hong Kong or Japan if you want to live long. Besides life expectancy, there are many other features in this dataset. The next one is mean years of schooling. Is this feature correlated with life expectancy? One way to check this is by looking at a scatter plot. I could find the scatter plot widget in my toolbox, but instead, let me show you how to introduce it to my workflow in a different, faster way. I'll drag an output link from the datasets widget and release the mouse somewhere on the empty part of the canvas. Orange now asks me what widget I want on the receiving end, and I can start typing scatter plot. Here it is. I click on its line and an instance of scatter plot widget comes up. Double-clicking on its icon by default shows me the first two features in my dataset, life expectancy and mean years of schooling. Looks like the two are related. The longer you go to school, the longer you live. It is not this simple. We should not equate correlation with causation. Yet, it is interesting to see that people live longer in the countries that better care about education. I now wonder which are the countries where people spend a long amount of time in school and have a high life expectancy. Let me select the data points that represent those countries in the scatter plot. In the workflow, the scatter plot widget can emit the information on its output as its dotted line is ready to be used. I would like to display the data selected in scatter plot in the spreadsheet. Therefore, I need another data table, and I will attach it to the output of the scatter plot by dragging a communication line from the scatter plot, and then from the list of widgets selecting data table. Double-clicking the data table 1 now shows the data of the countries I have selected in the scatter plot. These include Australia, Germany, and Canada. So if you like school, these are the countries you should take a closer look at. Which are the countries with shorter lasting education and also one with shorter life expectancy? We can select them in the scatter plot. See how the display in data table 1 changes immediately? There are also some countries where people attend school but life expectancy is low. let me select them in the scatter plot. There are Swaziland and Lesotho. We have just learned how to construct a simple workflow in Orange. In our workflow, we have loaded the data using the datasets widget, displayed the entire dataset in the data table, and explored the relationship between a pair of variables in the scatter plot. In another data table, we displayed the data on countries selected in the scatter plot. In the following video, I will show you how we can slightly change the workflow to choose the data in the data table and highlight the selection in the scatter plot. And I will talk about saving your work and loading the workflows into Orange.
All right. So I have a mock data. I don't know how to share with you the mock data. In WhatsApp or either email ID, common email ID, no? Okay. Aram, let's do one more thing. I hope you know how to use ChatGPT, right? So, okay, let me show you here. Yeah. So if we go to this Orange data mining, document here and load your data. Go to the main page, docs, and loading your data, and you will see an image here. We can convert this image into Excel sheet using ChatGPT. So I have, yes ma'am. Your ChatGPT will do anything that you ask.
Can I get anything from ChatGPT?
No. Oh, your app? No, go to the website. Yeah, go to the website. Yes. Orange data mining website. Yeah, data mining website. Are you there? Now this is app, ma'am. You go to the Google. Yes, ma'am. Here only. You go to docs. This one, yeah, docs, and loading your data. In there you see the image. Take a screenshot and ask ChatGPT to give you Excel sheet. It will give you. Where is the Excel sheet? I could not download it. Oh, okay. I could not. Yeah, you can take the sample. Okay, that's cool. Yeah, please get the sample Excel sheet and let's load into. Not opening?
It's not opening.
Okay, if it doesn't open, take a screenshot of that image and ask ChatGPT to extract data and give in an Excel sheet. ChatGPT or DeepSeek. Can you give me one bottle? Thank you. For this kind of small jobs, we use ChatGPT. So, loading your data. And this is, if you go scroll up, so this should open. If it doesn't open, let's take a screenshot of this one, and then ask ChatGPT to give you an Excel sheet.
Screenshot, no?
Yes, sir. Screenshot. I don't know. Okay, yes. Here it is. So you have to press the function button. This one. Yes, great. Yes, that's great. She's got it.
In ChatGPT or
Please extract the data from the image and give me in Excel format.
you can paste the
You can paste the image, copy and paste the image. It will take. Yeah, or you can take a print screen and then copy and paste. So print screen function. Yes, sir. No, sir. Print screen, sir. Function and so you can cut it and then you can use the mouse only to drag. Yeah. Select whatever you want. Yes. And then copy this, sir. Copy this and no, then copy this one. It's not copying. Let me try once. Yeah, it's saved here. Just copy this and go to ChatGPT and paste it here. Control V and ask, give me the data in Excel format. Then copy and paste in the Excel sheet. You can functions. Whenever you see the blue color, you see a function and then print screen.
Function and print.
Yes, ma'am. Ma'am, I will show you.
Okay.
So you can select, ma'am, this one. Select. Now it will be saved in your picture. Now you go here and you will see picture here, screenshot, and you have it here. You can copy it and go to Excel sheet. I mean, not Excel sheet, ChatGPT. Oh, so he has done a favor to all of us. He has uploaded that file in the in your in the WhatsApp group. That's good, ma'am. And that's fine, ma'am. You have a header and then you have a call. It's fine. You can. It must be there in the picture in the file. Ma'am, 1 minute. I'll show you, ma'am. So whatever that you took a screenshot, it must be here.
Pictures.
Yes. So now you copy this and you can paste on ChatGPT and ask it to give you an Excel format. Yes, sir. Sir, no, not this one, sir. So here, and this one, sir. Yes. So usually it's not downloading actually.
Making into copies, sir.
So you can take a screenshot like this.
Okay.
Yes, sir. It's just okay. It's placed here, and then
Excel sheet.
yes, sir. It will give you, sir. We have only 20 minutes more. It's not giving you. Okay. Okay. I'm giving you this way. Or we can do. We can copy this. Copy and put in Excel sheet. Okay. It's not giving column.
Same span.
You can directly open Excel sheet. Yes, sir. It's not giving. So what can we do? We can do one more thing. Data, text column. So it's come everywhere. Huh? Yeah, we can do this like this also. Okay, let's put it here somewhere, and we can save it somewhere. The document. Yes, I think we are ready. Okay, I hope you all have data. So let's bring the data here. You have the file here. Can you load your data into the tool? Say.
పేస్ట్ రావడం లేదు పేస్ట్
Yes, sir.
Sorry.
So these are different formats, sir. It's no problem. I think you might have pressed this one. Maybe double press. Yes. So you can. Okay, you have this file. Oh, you already have this file. Ah, okay. So this may not be. So the file one, you need to add the data. Yeah, go here. So you have this data. So where are the files are that you have? Excel file. Do you have Excel file? That he shared in the WhatsApp group. Do you have a file with you? Oh yeah, it's cool. Yes, yes. So next step is like, okay, you have the data here. Suppose if you want to see, maybe this one, then just bring it here and then do it here. Yes, sir, that one, sir. We don't want this one. We want visualization. Yeah. Okay, let's bring it here. That's not taking. Box. Yeah. Maybe it's not showing. So the correct format we need. Maybe we can try this. So we'll try one by one. I don't know. It says only two things. What are this one? So if you add a data table here, it will tell you.
Can I try this? What is?
It says G1. There are
Okay.
It made two groups. Just two groups it made. Yeah. Group 1, group 2, based on the mobile number and consent. So statistics makes things easier, right? So if you have 1,000 column and 1,000 rows, it will give you one picture, which will give you all the information. So that's the use of statistics and tools. So we are going to do the same thing. Okay, so what we do. So you have to find the table, then you start your analysis. So analysis, you can see here. Yeah, so here maybe we can go scatter plot, right? You move it here, then connect them. So we need to select the appropriate statistics model with what we want to do. Maybe let's see this one. Okay, yeah. Thank you. Okay, maybe we can tell it again. Maybe scatter plot. Oh, cool. So the scatter plot has taken 3 parameters: protease, respiration, and ribosome. So based on this 3 category, it has made okay, I don't know why it is moving here. Then if you want to see what is that, again you have to connect to the table. I'll show you. So you have this one. So now you connect, you bring the table again here, and then you connect this and this. Suppose if you select this, it will tell you in the database.
Yes.
Right? Yes. So
Empty sets.
let's delete this one. Let's move this to oh, sorry. Is it saved, man? I think saved. Now go here and you have to move your file here first, then go here. Where is the file, man?
Desktop.
Yes, desktop.
Desktop.
This one?
That one.
Okay. Now you bring a data table and connect these two, and it will tell you. Right? You have the data now. Now, how do you want to visualize the data? So that is up to you. So you can select either box plot or violin plot or scatter plot, whatever you want. So make a connection between from file to your scatter plot. See, it's going to show you. So it has categorized 3 categories: protease, respiration, and ribosome. Right. So based on the early or mid or late. Yes. So I think this is early. I really don't know what is this data actually. But if we have a yes, ma'am. It's just a visualization. Yes. You can visualize with in many form. You can maybe visualize. Okay.
Radial form.
Yes. You can
Bar plots.
not. Maybe let's see if we can visualize this. Yeah, actually, this is not.
This one is good.
Yes, ma'am. Maybe this one. Maybe this one. This is Venn diagram. Oh no. Or heat map we can give.
Then there are not common sharing factors. That's why Venn diagram is not coming.
Yeah. So I think this is about gene expression. That's gene expression. So the best way to see is heat map. Okay. Can you please select the heat map and then observe your data, how it looks. Ma'am, can you close that one? Close this. And okay, you have to connect it directly to the file, not
Data table.
not through the data table.
Again,
Yes, ma'am. Only the ma'am. Can you give me that? Yeah, so let's connect it. I think this mouse is.
yes, it's very risky.
No, it's not showing anything. Why it's not showing anything? Ma'am, you have to make a column. There's no column actually. Wait, I will show you how to make the column. It's in the file. There's the file, the Excel file. You can open the previous file. Sir, see the heat map, sir. Can you go to this one? So you see there's no column here. So only one column, right? So here is the data pages, you get data, text column. Okay, next. So you see each one of them is there's a comma, right? So wherever there's a comma, yeah, wherever there's a comma, we can make a column. So you see here, it's a different column. Input. Now if you go, it will come. Okay. Yes. So you can't. So depending on the data type. So G expression should always be visualized in a heat map, so you can easily see.
Now we are using this file, data table, data set.
That's a big. If you have another data set, no, it's just like another file only. So if you click here, you can click. Let's open this. It's going to have,
From here we can
yeah,
select.
it's going to have same columns or whatever. Actually, the big data maybe we will see tomorrow. I don't know, tomorrow we have lab. Big data will have hundreds of columns, rows, text column, and delimited. Okay, next, and click the comma. Yeah. So basically, you are telling that wherever there's a comma,
Yes, sir.
there is no comma here. Okay, if there's no comma here, you should select space. You can deselect the comma, right? So wherever you see a comma or space, that we are asking the Excel sheet to make a column.
Text column, next comma, sir, paste in answer.
Yeah. Finish.
Sir, first text to column,
Data.
Data, text to column, and then you can click, sir, no problem.
next.
Yeah, it should be always delimited. Okay. Then next, then you have to select where you want. Sometimes it is column separated. The data with this. No, it's very simple, ma'am. I'll tell you. So this issue.
Search a picture.
Okay, ma'am. So we can, let's move it up here. So we'll select this one. So it is in only one column, right? So our aim is to make it in a different column.
Pasted in one column.
So we'll select this one and go to data, right? Text to column, because there's only one text, we are making them into column, text to column. So it should be always delimited. Then go next. So wherever there's a comma, we are going to make a column. So we have to select here. Okay. So then it will become a different column. See? Wherever there was a comma, it made into a like.
CSV to Excel format not suitable for this one.
It should work for me.
CSV file also.
Yeah, CSV, yeah.
That CSV file, comma separated, that can be converted into like this.
Yes. Yes. Yes, ma'am. Yes. So I think we are almost done. So far we have seen yes. So far we have seen the importance of disease, its role in yield loss, and causal agents, and then teachable machine, how to train particular model, and then we looked at all the steps. Then Plantix app. This app is easy. Like you can ask all your farmers, relatives to use it, and then we looked at the orange data mining and visualization, right? So each data has its own particular way to visualize. Okay. So in case of gene expression data, heat map is the best one. So it will give you a shade of color, right, from higher to lower. They said 250 that we should leave to see the central instrumentation facility at the Department of Plant Sciences and School of Life Sciences. Right. Sir,
It will be there.
I will be with you, sir. So let me go and fetch some. So tomorrow mostly it will be theory.
covered the trying to cover? What is the what constitutes doing? Main for identification of identity. Platform is there where the group of people, platform is there where the group of people, platform is there where how many of you teach platform? Because we have enough filters to teach, but we know the students are that. Maybe because it's our fault, maybe teach it to the students, and we don't give the importance of platform. Identity we use for identification of class. So that's the main thing what is required for biodiversity conservation. That's what's lacking, and that's where AI is filling the gap. Usually the students go to the field and try to plant the animals and analyze what they do. I will be talking about giving you some fundamental ideas about whatever you know about especially the species. Give you some introduction before you start. I think it should be. How conservation what is the species? You think of fundamental unit of and maybe why we need to find out what because it's showing biodiversity. Also understanding evolution, how and usually this aspect is so far going back to the how will studying all these things, just brushing your memory on this. So appreciation belongs to diversity without this part. Study process of appreciation. There's a word called synthesis. Games or other kinds of appreciation of your other things this is part going forward that or green part these parts are shaping their forward and this part I am not necessarily when I not go to so I because this part have to to grow on what I call secretary cross. Secretary cross for the irrigation, so these plants now they have some mutations which they will grow. So this plant that are growing this area now forms a genetic barrier. So these plants are not cross these plants. So they are specialized. But it's kind of they call the ecotypes. Specialized in over a period of time, now they are the same species, over a period of time, so this is the example of these two class you can see one is species Abby versus the other one is Sigua. Sigua is widespread in Japan. The area small population morphologically they have some characters like seed name is so they But suddenly these guys, one of the population, they could self-fertilize. So now they don't need of any cross-fertilization, and they became isolated, genetically isolated, and slowly they will become into a new species. So this is a classic example of a symbiotic speciation, you can say, one small mutation which can change it into a new species. And another way is, another example is this one, monkey flower. You see these two flowers are completely different, mimulus, different species, Lewisi and cardinalis. And what you see here, this plant is having a pink color flower and it has a yellow landing strip where it can accommodate the pollinators, which is a bumblebee. Bumblebee requires nectar, but it requires concentrated nectar with low quantity of nectar is enough for this plant. But this one, usually when you see a red color flower, that means it's either pollinated by a butterfly or it is pollinated by a bird. So in this case, it is hummingbird pollinated. It is bright red, it's attracting the hummingbird, and it produces copious nectar. So it is providing more nectar for its pollinator. So what scientists did where they plant, these plants grow very nearby, they flower at the same time, but they don't fertilize, they don't cross fertilize. They brought it to the lab and cross fertilized. They could form hybrids, and they could form fertile hybrids. These hybrids also could back cross with them or cross among themselves, breed among themselves. So this is a case of a biological species. Biological species concept, Ernest Mayer's biological species concept says that there should be a reproductive barrier, a group of individuals, a group of population which cannot interbreed among themselves. That is a species, right? But if you consider these two as different species, they are now interbreeding. Now your definition of biological species is not worth here. So you can say this is another way of speciation where certain changes in some parts of the chromosome have changed themselves, and it has been adapted or selected by different kind of pollinators. One was selected by the bumblebee pollinator, one was selected by the hummingbird pollinator. And in such cases, you get some what we call as rapid radiation, where more species can be evolved within a short period of time. That's what we call as radiation, rapid radiation or radiation. This is a clear example of what we see with Darwin's finches also, adaptive radiation. Where each of these, this is also kind of ecological speciation. Each bird found a different ecological niche, or it found a different food source in different island, and it specialized in that area. And then that's how immediately you found, within a short period of time, at least 25 different species evolved. So this is how speciation occurs and how biological diversity occurs. And coming to taxonomy, taxonomy is the art of naming, a system of naming the plant so that we can easily identify the biological diversity. And you know, which is a rank-based system. You have different ranks like the kingdom, phylum, class, order, family, genus. All this is a fixed rank-based system. But there is another thing called the cladistic or phylogenetic classification, where organisms are classified based on clades. So clades are a concept of the phylogenetic tree. The phylogenetic tree is a tree diagram which can depict the path of evolution. So like your normal tree, your phylogenetic tree also has the leaf, the branches, nodes, stems, and the root and all. So this is just showing the how evolution works. So we all know we all have a common ancestor. We know we all share the same kind of genetic material. We all have the same amino acids which make our proteins. So we know we are all interrelated, and we are some way interrelated to the most common ancestor, which is at the root. And the phylogenetic tree diagram is going to represent that evolutionary pattern here, where your species or genus forms these branches, and the ancestral branch of these two species is indicated by the node here. That means there was an ancestor here which gave rise to two different species. And along the branches you see these kind of dots which are nothing but synapomorphies, or new characters which are derived across the lineages, in those lineages, which can be used for classification here. So when in terms of cladistic taxonomy or phylogenetic taxonomy, instead of forming this kind of rank-based system, people now go to use clades, what we call as clades here. So even if you see now in the Wikipedia, you can see each of these groupings are now called clades, eudicots, rosids, like that. So this was a different kind of taxonomy which was proposed in 2004 based on the idea of evolutionary biology, but still it is not taken into consideration. So molecular phylogenetics is the area where we use DNA sequences. You collect DNA sequences and try to map them in a phylogenetic tree here. So that is another method. So you can relate molecular phylogenetics to computer science, where initially people use morphological data for understanding the relationships here, right? But here now people have started shifted from this morphological based taxonomy. This happened in the 1950s itself in the science called cladistics. You might have heard about cladistics, where people use morphological data. Instead of assigning the taxa themselves, they give some computer algorithm which can trace back the evolution. So for example, this character here, T, is shared among species 1 and 2. That means the ancestral region was probably A here, and these two species have derived a new character there. So that in that way you can say these are recently diverse species. So here I am using the DNA sequence as information here, but initially they use morphological characters. When you're classifying insects or birds or anything, you take all the morphological characters as a matrix, zeros and ones, and then you feed it into a computer, and it can make trees like this, based on UPGMA, neighbor joining, parsimony, maximum likelihood of Bayesian. These are all applications nowadays we use with computers, again using intensive computational work to build robust phylogenetic trees which are most reliable nowadays based on the data we give. And here the data what we give is genetic information only. And now we know what is a species, and you have heard about something called cryptic species. So I will just briefly say biodiversity also includes cryptic species. So species which cannot be clearly defined by morphology. Those are called cryptic species. I will give you some example to give you an understanding how biodiversity exists in the form of cryptic species. So this is the grey langur or the Hanuman langur. This is a very common one. Have you seen this in your? It is all in the entire peninsular India. This the macaque is more common, but the langur is also common. It's common to the peninsular India. Throughout India, you will see this monkey here. So it's found in southwest India and in Sri Lanka, and it was a monotypic genus, meaning that only one species was there in this genus, Semnopithecus sentalis. So this is what Nilgiri langur Trachypithecus, and this is a purple-faced langur. It is found in Sri Lanka. It's called Trachypithecus velutus. So these are all other Trachypithecus which are found in Southeast Asia. In Arunachal and all these places you find these kind of monkeys. So I have introduced one genus called Semnopithecus, and the rest of them are Trachypithecus. So when you are doing a phylogeny here using a DNA sequence like I mentioned before, you can see the Hanuman langur, Nilgiri langur. So Nilgiri langur was a genus of Trachypithecus, but it is coming, or it is showing a common ancestry with the Hanuman langurs here. So if you say this node is indicating a point where there was a common ancestor in which the lineage is split, you can see this is this clade includes the Trachypithecus and the Semnopithecus are coming together. I mentioned that Semnopithecus is a genus, Nilgiri langur belongs to Trachypithecus. They are two different genera, but when you are building a phylogenetic tree, it is saying that they are more closely related. So what we should understand that by using a different kind of data set, we are finding that the morphological characters which was used to define Semnopithecus is not good enough to say it is a different species. So a different genus here in this case. So if your mitochondria says they are belonging to a different genus here. Yes, this is based on molecular phylogeny, and this is based on morphology. I'm just naming the clades based on the morphology. So this is what we call it as cryptic species. Many times you don't know whether to call it to that genus or that species because it doesn't have enough morphological characters to separate them. So we initially told that species is the final entity of
classification. But within a species, there could be a lot of variation also. And that could not be captured, and that can be well captured with molecular data rather than morphological data. So where the classification is not subjective, when you're using DNA data, it will become objective. The DNA will give you the answer, and then you can understand. Even though the species are very recently diverged, it won't have enough morphological characters to show them as very different. And another thing is they could interbreed among themselves also. The interbreeding could also happen. But in most cases, we cannot find out whether it is a species or not unless you do some experiments. Like you can't use the species concept as Ernest Mayr, whether they are interbreeding entities or not. You have to bring it to the lab and then do interbreeding and see whether they are different. But that's not possible. So that's why we just monitor the biodiversity just based on morphology. And this is what a better explanation of a cryptic species is. So cryptic species means the species, ancestral species dividing into two different species. This could be the separation of the reproductive traits, molecular divergence also, that means how the DNA sequence is also diverging, and the morphological divergence also, and also the behavioral divergence and ecological divergence. So that each descendant occupying a different ecological niche. They have to separate in all the different ways of differentiation, but here it doesn't happen. In a daughter species, if you are looking at one of the axes, if you are looking at molecular divergence, probably it has not diverged in that axis, but it has diverged in other axes. So those are called cryptic species. So if a morphologist is looking at morphology has not diverged, but its DNA has diverged. In that case, it is a cryptic species. It gives more confusion. So now our idea of species itself is also kind of murky. You can't clearly define a species. There are more young species, you can say, they are in the process of diverging, and still they can interbreed among themselves also. Unless a very strong biological barrier is formed and over a period of time, only then they will become different enough. But when you encounter biodiversity, this is also a different kind of biodiversity you will encounter, which it also should be conserved. And that's what we study at the population level, that biodiversity at the population level. So I will skip all these things. So how to deal with biological, this kind of cryptic species is you have to use different methods of distinguishing them. So that is what we call it as integrative taxonomy. Traditional taxonomy uses only morphology, whereas integrative taxonomy uses different things like molecular data, studying the ecology of the individual, studying the behavior of the individual, many things. So you can see integrative taxonomy uses MSc is morphological species concept, BSc is biological species concept, PSc is phylogenetic species concept, which is based on this building phylogenetic trees using genetic data. So non-morphological based axis is your ecological axis, behavioral axis, and molecular data. Axis is, for example, there was a species of crickets in IAC, they recently published it, based on the call sound itself. The call, each species has its, it's a waveform of its call, right? There's a particular waveform. So based on its unique waveform also, they classified, they named the species. They use it as a criteria to define it as a species. So you can also use the ecological characteristics for the species. Mostly molecular data is very convenient because the data you can collect lots of data and then use it to separate it as a data and then use it to separate it as a species there. And next comes the of extinction. So many of these species what we see today, which was developed due to this kind of speciation processes, including the cryptic species, are under threat. So for example, these the whale here and the panda here and the small animals like frogs and even these kind of naiad, which is kind of a mussel, all of these are all under threat. These are all endangered organisms. They have not put plants here. They will also be more. Insects are lots and lots. And many of our the closest relatives of the humans, the gorilla, the bonobo, orangutan, all of these are now considered as under threat. and including this very rare vaquita, which is kind of a dolphin, is critically endangered, which estimates ranging from 10 to 18 individuals only remaining in the wild, because due to fishing, unwantedly they go and get caught in the gill nets and then they die. So these are all what we are facing because of all the diversity what we have seen here. So how to overcome that? So the red list of threatened species, this is a kind of database where you have all the IUCN red list. So each year it updates the red list, and now there are about, in 2006, there was about 7,025 species which are indicated in the red list. And this is a very small number, and only certain species are studied for their vulnerability, not all. And then what you can see in the red list website, IUCN's website, what percentage of each individual or each animal has been under threat. Amphibians, 41% of the amphibians, known amphibians are under threat. 26% of the mammals are under threat. The 34% of the conifers are under threat. 12% of the birds are under threat. Sharks and rays, 37% are under threat. Coral reefs, 36% are under threat. Cycads, the ancient primitive gymnosperms, 71% of them are under threat. Reptiles, they don't have data for insects here. They will also be under threat. And they have not classified the flowering plants. They have given only the cycas here and also the conifers only. So all of the organisms are under threat now. Well, it's known there. So what is the definition? How do they define these? Does anyone know how the definition of these, how they are assessed for threats? There are different criteria. So, and also they are given as IUCN status, site status, national status at different levels. They are assessed for their vulnerability, whether the population is going down or going up. I mean, of course, most of the populations are going down. So you have these different categories you might be knowing, right? This is data deficient about the species. That means the biology of the species might be well known, the morphology, everything will be well known, but it is not studied for the threat. That means it is how it is going down, its population is going down or not. That is not studied. So that is called data deficient. So data deficient is doesn't mean it's not under threat. It only means that we have not studied that organism for that threat status. And then you have the widespread and abundant taxa. It's the least concerned one. Not most of the plants, all the VD plants what you see around are under least concern only. And then you have the near threatened. It has been evaluated against the criteria and don't qualify as critically endangered. And the next one is they are called vulnerable. Is considered to be facing high risk of extinction in the wild. And taxon is endangered and considered to be facing very high risk of extinction in the wild. This is endangered. High risk, very high risk, and critically endangered, facing an extremely high risk of extinction in the wild. Based on the criteria A to E in the section individuals, now it's only 50. That means it is under vulnerable status. Now you find only 90, then that means they are critically endangered. And another way to assess is based on the geographic range. How much of area they are covering, how much of forest area they are covering. Two important criteria they have, it is EOO and AOO. Area of extent of occurrence, area of occupancy. How large area these plants are growing, plants or animals are growing. If it's critically endangered, they are growing within only less than 100 square kilometers only their ranges. They are found only there. That is endemic to that place. If you leave that place, if you go to, they will be in some patches, right? If you assess that area, it will be only 10 square kilometers. They are found only in 10 square kilometers. That is also, so that also means they are critically endangered. So these are the main criteria how they are assessed for the endangerment. This is the most common way to find out whether the species is endangered or not. And apart from that, there are number of mature individuals, very small restricted population, all are the other criteria. If there are very mature individuals are less than 250, then again they are considered as critically endangered. Number of mature individuals, that means again they are considered endangered here. And any other analysis methods also they use. So these, based on these criteria A to E, 5 criteria are there. In any of these criteria you assess them as endangered or vulnerable or threatened. So the next is about the national status. Some of the, apart from IUCN, other agencies also can maintain this. Like some of the states have their own threat status for some of the amphibian species. Like for example, in India, these are the endangered animals: the Himalayan brown bear, red bear, pygmy hog, Andaman white-toothed shrew, Kondana rat. All these are critically endangered mammals here. And then you have the Elvira rat, Namdapha flying squirrel, Malabar civet, Sumatran rhinoceros, Kashmir stag, and the marine mammals, Ganges river dolphin, Indus river dolphin. All these are endangered. And those are critically endangered. These are the endangered mammals: Asiatic wild dog, El's deer or the brow antlered deer, golden langur, which I showed there, it's in the Nilgiris, Himalayan white-bellied or musk deer, hispid hare, hog deer, lion-tailed macaque. These are all the endangered mammals here. Nilgiri tahr, red panda, cheetah, and all those things. This is a kind of a Tibetan antelope. So again, within the state also, within the national level also, you have some endangered list. Within the state also, you have some list here. So for example, in Kerala, there is a species called Nasica batrachus, is endangered for the pink colored or purple nosed frog. The frog looks purple in color. So the area which that frog is there is also protected under conservation acts. So that is regionally they are having this different kind of rules. So these are all different measures what we use to conserve the animals just by finding out their threat status. So we talked about a little bit of endemism also. Endemism means the plant or animal is growing only in that area. So that is also an important criterion showing the biodiversity of that area. We'll just skip that. And coming to the how to conserve them, how AI can help in conservation. Now we have known whether they are endangered or not, and how they can be conserved. And most important is the, I will give you some examples here. And there was one project called the Snapshot Serengeti. So this Serengeti is a place and is well known in National Geographic. Very often they show documentaries about this, right? Where the lions, elephants, giraffes, all these wildebeest, all of them are there. They're in the there's a huge assemblages of animals are there, and even in these places they are not free from threat. They are facing a lot of threat. How people can conserve these things? How conservation biologists can work? So the main work of biology itself, or the main application of studying biodiversity itself, is for conservation, right? So how to conserve this? And a very good example was in the Serengeti. I think this was long back, maybe 5-6 years ago. So this is mainly using machine learning. You saw about, I think Professor Srinivas, he talked about how to differentiate a animal, a cat from a dog, right? How machine learning algorithms work. So they define the images in one particular format, and I think Madam talked about a format called JSON format, where you collect all the data and store it in a digital format. So all the characteristics of a dog will be given in one digital file, and a cat will be, so the computer can differentiate between a dog and a cat, and that's where the most important aim of machine learning here, the most important use of machine learning in conservation biology is here. So in the Snapshot Serengeti, they could get around 3.2 million photographs from the Serengeti national park here. So what they do is easily, this is consider this is the Serengeti forest or any forest which we want to conserve. They use camera traps, mainly used for conservation. They use these camera traps. They constantly collect data. Initially, what conservationists used to do is, you have to have a role of rangers the government, even now we have forest rangers, they go into the forest every day and see whether all the animals are healthy or not. That's how we manually do it, right? So when the area is big, you have to spend more money for the rangers, and even how many ever rangers you put, they can't cover the entire forest. So now we automate it using these kind of camera traps. All forests, even our Telangana Forest Department, Andhra Forest, every forest department in India have this camera traps as a very good or very important tool for their conservation efforts here. And these are heat activated motion sensors are there. When the animal comes near, it can detect that and take pictures, otherwise it can't completely take. So, and these can be left for maybe 1 month in the field. You have enough charge in it and then leave it for 1 month in the field, and then you get these kind of snapshots. These animals keep moving, coming here and there. So the main problem is data analysis with this. So now we have got machines to do the work. The next also we are faced with challenges. So camera traps, you leave it for 1 month, you get around 1.5 lakh images. So how to deal with it? You have to put so many students or staff on this to analyze the data, right? First you want to know how many animals are there in the forest. So that means more snapshots are there, that means more animals are there, then you know the population is healthy. But getting that is the most difficult problem, and most problem what they faced was many times nothing will be there. Sometimes the camera would fire by mistake, or maybe some tail will be there, you don't know what animal is there, you can't identify that. So that's why you have the AI model. You train the AI model to identify the animal very easily. Even a tail portion is there, now your model is well trained. If it has a stripes or something, you should be able to capture that. So I think for machine learning, they tag the images. If it's a tiger, they tag it for several times, they train it and then use it for, and you collect it as a data set. And then the most important help AI model could do and identify animals, but at least it can identify no animals. It can just eliminate. It will take so much, it will help, do a big help. If 1.5 lakh images, maybe only 5,000 were having animal photos. All of them has to be eliminated. If somebody sits there and does the job, it will take, I mean, several reports say that it will take 6 months to screen this much of this, and if you are employing 5-10 people also. And it will become more expensive, right? If you are employing 100 people to work on this, it will become very, very expensive. So this is where really AI has helped in doing this. And now it can identify the animals. Yes, animals are there. So this is itself a big task, removing the false thing itself. And then you screen only the things which have animals, and then you can get more data also out of it. Which species, you can classify how many species of animals are there, how many of them. If you are frequently seeing in the photographs, you know their population is slightly higher. Then you can also calculate their age, sex, if the photos can distinguish the male and female, and also based on the body size, you can calculate their age. All the data of the forest you can continuously monitor using these AI tools, instead of using the human people. I mean, if you employ few rangers, the government can employ only few people, right? You cannot get that much of data, but these machines can do that much more you can get. And also you can find out what is the community structure. So population is a group of individuals, community is a mix of species, multiple species interacting among themselves. How many individuals are there in the community, all that you can find out. Eventually your aim of conservation is being achieved here, and you can How are you determining the species? Cryptic species you cannot hear. Cryptic species you have to go for other, genetics would be a better tool. Yeah, that is also part of AI, you can say, because it's using big data. Again, you're going to use big data, automation, everything. Cryptic, definitely you cannot do it here. This is mainly for large mammals. Cryptic speciation is more in insects. In mammals also it will be there. Now you know about this cobra, right? Initially cobra was only one species. Recently science work from IAC, they proved there are 3 different species of cobra. Something in Indonesia is different, one different in northeast, the southern one, Western Ghats one is different, the cobra itself. What's that? Naja, yeah. But now they made it into Ophiphagus, right? Ophiphagus. King cobra, yeah, I was talking about king cobra. Yeah. So that was also a cryptic species, but they used genetic data only. Once you find the genetic data, you should also corroborate with the morphological data. People will do that. If you do a deeper study, you do morphometry, you can also use ML there also. But if your data size is big, very big, but for snakes and all, you're collecting the king cobra will be very less only. Max catch them, their population also will be very less. So there you cannot use it. But this is mainly for this Serengeti and all those places. You can use this. And you can also know the community structure. How many individuals are there? How many different species are there? How many mixed species are there? All that you can find out. So a lot of examples are there. So again, there's something called this. This was done in Serengeti, the one I showed, but also it was done in Colombia, another the South American forest there. So there's wildlifeinsights.org, which is Google AI's project there, where they used the same technology. It was, I think, Serengeti is a kind of a grassland. It's not many trees are there, but the Colombian forests are kind of wet evergreen forests. There, they also use the camera traps to study that. So what they say is they transfer Google, and this was supported by Microsoft, this was supported by Google. They use their own cloud services to, they also, as a experimental approach, they give free access to the researchers there. They immediately transfer all the data from the camera traps to the cloud, and then from the cloud they will develop some APIs which can do all the processing from where it is in the cloud. They need not process the data in the ground level itself. And there was some project called the Hakuna Matata. That means I told Serengeti had lot of the millions of photographs. To analyze that, the best machine learning approach, they kept some competition where each team can find out their own algorithm to screen, or they can develop their ML tools better. I mean, in machine learning, how to eliminate the false positives or how to make better hits. So there was some, and that was about on the Serengeti data, and they gave a prize of around $12,000. That information is there about on this website. And this is about the snapshot Serengeti. Here, what they used here, and finally what they got, they mentioned here, and it is listed in this website called Label Information Library of Alexandria, which is a kind of a database or a website which hosts these kind of different projects, mainly machine learning projects for each specific project here. And this is called Leela BC observation. So the one thing you should note about here is Serengeti National Park is around 30,000 square kilometers. The core area is much smaller, maybe 15,000 only, but surrounding and all other patched forests, everything you cover, it's about 30,000 square kilometers. In comparison, you have our Western Ghats, it is 160,000 square kilometers. So you have wide application if you want to do it in India. But we don't have such a big project, well reputed project here, but now people have started using machine learning approaches also for our data sets also. I mean, the amount of camera traps you have to install and all is there. But now all forest departments have camera traps to monitor wildlife continuously. It's better than using manual labor, using rangers, forest rangers to go and check those things. So like that, a lot of examples are there. One is the community fish data set, fish detection data set. And there's something called the shark spy. This is in New Zealand, where they use some, instead of, this is an aquatic system, right? So they use some bait here, and then they keep a camera. Maybe it's a fixed system where there's a bait and a camera attached to it. And it records for 1 hour, it seems. And after that, the computer detects some motion there, and then it makes these small clips of around 5 seconds, 10 seconds. And then then they make this publicly available to others. So this is what we call it as citizen science, where everybody participates there. This is very popular in US and other countries there. In India also there is one Season Watch. Have you heard about that? They are also very active in colleges to study the phenology of plants, at what times they flower. That's a very well-known, the most popular citizen science project. Apart from that, India, we collaborated with several other citizen projects like iNaturalist. It's a well-known one. So here you can check your, I mean, they allow you to see the video, and then they give you a morphology of different sharks also. If you can identify which shark is that, you are training the model there. You're validating, giving your input there. This is one of them, and other one is the Ohio Small Animals project here, where they had labeled many things, and this, as the name suggests, it is for small animals. It is not for the mammals like in the Serengeti. They trained it to identify the snake, song sparrow, meadow vole, and blank. These are all different blank images. So they were trained to find out. These many images were used to train based on the data set. Now that data set is also available. You can go down and find out that data can be downloaded here. COCO. Have you heard about this word COCO? It is a Microsoft's format for, it's a JSON format where all the data characteristic of that fish or that particular animal here, meadow vole, everything is in that format. You can download that, and if you want to use it for your data, I mean, this is a sample model, you can say, ML model is there. You can use your camera trap images to use that model to identify your images. All of them are publicly available. Likewise, you have these again MIT Sea Grant river herring. This is for the sea grant. That means that some grant which was given to the sea research, and mainly they were researching on this one, the river herring, a kind of fish there. Apart from the fish, they also did this video clips only instead of these camera traps, like how they study the sharks. They captured the video and then they shared the video among themselves and used machine learning to identify many things. Mainly it was 80% was annotated for only the herring, but other fishes were also trout, bass, perch, eel, sunfish, lamprey. All of these were also found in the community, and that was also trained, and that data can also be downloaded now, also in the COCO format. And again, community fish detection data set I showed there. Images, they have analyzed around 1.9 million images, and these many bounding boxes, and each 17 individual data sets spawning freshwater, marine, and lab-based captures. And this is also a well-known database which can be, which they train the ML and is publicly available for you to use it. But mostly it will work for only that project, that region only. You can't use that project on Indian systems because those animals will not be there. But you can get some data. If you're able to write code, you can also use that data here also, or at least you can find how that code works. As a study method also you can use that. And I just briefly want to talk about diversity itself. What is alpha diversity, beta diversity? You heard about it, right? Just to make you recollect, we talked about the community structure. So this, for example, if you're doing a field work on one, usually we do it on small grids, like quadrats. We do quadrats for trees. We do bigger quadrats for plants. We do very small quadrats. I mean, herbaceous plants, we use 1 into 1 meter quadrats. So like that, it can be called as a site one. So how much of the species are there is called alpha diversity. Likewise, if you are studying multiple quadrats, so each one is a different site. So each one has a diversity. Here, how many species you have? 1, 2, 3, 4, 5. 5 different species. That means it has an alpha diversity of 5. So next comes a beta diversity. This is alpha diversity. This comes, so say you're looking at 2 different sites, I mean 2 different national parks, and these sites are referring to quadrats inside a national park. Maybe it is within a state or within the southern part of India. And beta diversity is comparing the difference between these two. How many are unique, or how many differences are made, how this site 1 is different from the site 2. How many different species are there in site 1 and site 2 is a beta diversity, studying the beta diversity. And then you have gamma diversity, which is nothing but the entire biodiversity of the area. Maybe you can consider for the entire state or a country also, you can say. So diversity can be measured at different levels, and these are different measures of measuring the diversity. And now the sites, I told about sites, right? this is nothing but the Convention on International Trade and Endangered Species. It is like a part of IUCN, where trade on many of these animal or plant derived parts are banned. Nowadays, elephant tusk is completely banned. You cannot sell in the market. It doesn't have a value. So many of the things are listed in the sites. So this is a kind of a database which can be also used through AI for conservation, which I will talk next. So what is the conservation? So we'll just recollect what we saw here. So what are the threats animals faces in the wild and how we can mitigate them? First is mainly habitat loss, poaching, what else? Overexploitation, disease also, climate change, pollution, forest fires, all of them. So in all of these, habitat loss, animal migrations, human-animal conflict, this one we left, animal trade, what we talked about, the CITES, fire. So these are some of the things. So in each of these, using AI can be helpful here. Mainly for poaching, they are nowadays using drone mapping. So instead of we use camera traps just to find out the community structure and what is the population, age of the individuals, of the species, all that can be found out. But nowadays people use this, instead of people going and patrolling, you have these drones which go for extensive patrolling, because it's huge forest areas, right? Even the rangers will have a separate path, but the poachers know the other paths. So they know how to avoid these things. So that's why people nowadays use drones there. Now, all of these have integrations with AI. So drones can identify some abnormal activity. Some, even the drone imagery can be screened with ML, machine learning can detect any abnormal activity, people moving around in the drone images. Then the drones will be designed, the drone videos will be designed just to see the flora and the animals only. But you see some animal inside there, what time they were observed, and then use the geolocation, you can go and stop poaching. And also you can study about animal migrations. This is it's a very important phenomenon, this wildebeest and all these large populations of animals in seek of water. Sometimes some of the lakes will dry out, so they will move in huge patches to the other areas. So that time also you might find the poachers are following them, or disease might be there, like some disease might be attacking them. So all this could be identified using the AI technology there. And also human-animal conflict also, like where how the animals are moving inside the places, living places. And also you have AI models where you can find out, instead of people searching it, we can also give some key terms where how these banned products are being sold in the market. The AI can trawl around the web and then find out any tusk or anything is being sold. Instead of people going and doing it, they're using AI to find out animal trade also. And fire, of course, is a very important phenomenon. It's a natural ecological phenomenon. And very easily it can be traced using the satellite imagery. And again, this is an example of DeepMind's Alpha Earth, which is having a very huge amount of satellite information. This is from Google. This is a new AI model, integrates petabytes of Earth's observation data to generate unified data representation that revolutionizes global mapping and monitoring. So fire and all can easily be found out by these kind of methods. Microsoft also has a lot of these kind of data available. So a postdoc in my lab, she's working on this only. She's using geoinformatics to find out how vegetation changes occur. In NRSC here also in Hyderabad, ISRO's NRSC, National Remote Sensing Agency, they use this kind of, they are expert on this. Now they monitor the entire Eastern Ghats for these kind of variables. Now, the postdoc is also experienced in monitoring fire, how the fire has happened before, or any ecological factors which can induce the fire. We can predict that also, and we can be ready. We can't stop the fire, but at least to minimize the impact, we can do that. So for that, all this geo information is very, very useful. So geo AI has many applications. So geospatial AI, geospatial AI has real estate market trend analysis, customer intelligence, public health analysis, logistics route planning, agricultural crop yield optimization, finding mineral deposits, renewable energy potential mapping, sustainable city planning. So far, none of them have directly impact to the topic which I'm talking about. You have fire risk management, water resource management, wildlife habitat conservation, tree canopy mapping. So at least these four are directly related to biodiversity conservation. Also, sustainable city planning also can be related to somewhat to biodiversity conservation. But throughout, they all have a very important application for many things. Like public health analysis is also very important. You can use how disease occurs, mainly in humans also, you can see how it occurs. Logistics, route planning, all those things. Agricultural crop yield. So this covers the entire sustainable development goals, you can say. You have the 17 sustainable development goals, right? So geoinformatics has application everywhere. Geoinformatics or geo AI. But most importantly, these things: fire risk assessment, water resource management. Mainly the animals are doing this mass migration. It might be due to some water scarcity, and during the path, if they are not facing water, they might die. Lot of deaths could happen. So at least we can predict and provide water there. And also, if we can, you have these fire mitigation, right? So you can, the forest department can burn certain areas so that fire will not spread. So we can predict that, and then we can do that also. And of course, habitat conservation, like how they are doing it in Serengeti, and tree canopy mapping. This is very important. So forest deforestation is occurring. You can't go and monitor at the ground level. It is very difficult. Only with the satellite imagery, you can say, like 2020 it was like that, 2010 it was like that, 2025 it is like this. So you can clearly show how much of tree loss or canopy cover has been lost. So we can plan for more plantations, or we can stop. Only first we can prove that there's a loss of tree canopy. You can easily do that. This kind of work is being done in my lab. The postdoc is mainly concentrating on fire risk assessment and tree canopy mapping. We have chosen an area in Eastern Ghats. So we are monitoring the forest cover in that area, how it has been changing from the last 50 years, how we can predict it. And then coming to the next is how to identify the plants and animals. So it's a very important direct application, though it's not a huge project. So Audubon Bird Society is a conservation society so they have an app which can identify over 600 bird species. You can download this app and do it here. And most important is the eBird. Anybody interested in birding will be knowing about this eBird, also from the Cornell Lab of Ornithology. So this is also like your iNaturalist. Any bird photo you take, we upload that, what day it was found, the geolocation where it was found, everything. You can upload the photos also. So now whatever data we are giving will be used for a collective database as a citizen science program. So, and another one is the Merlin Bird ID. This is also identifying the birds, again from the Cornell Ornithology Lab. So this is a very popular app where every bird sound also you can play. If you can just hear the bird, sometimes in the forest it's very difficult to spot the bird, right? If you can just hear the sound, you can record it, you can upload it and find the match, or you can hear the other birds also as a sample birds. A different sample for each bird species. What would be the bird sound call? All of that will be given here. So for birds identifying, apart from the morphology, the call is also very important. So that database is present here. And coming to plant identification, can anyone identify this plant? Which one? Butilan, yes. Or yes. So this one I collected here, but if you are giving, see, this photo is not very clear. You are very familiar. You found it, even only the flower is very small, but still you found out very clearly. But I mean, the ideal thing what we do is the best. Our day-to-day what we use in AI is even before the advent of AI, this Google Lens was there, right? If you don't know anything, any product or anything, we just upload the photo and find out. So I tried to identify it using ChatGPT. It gave a wrong identification. Oxalis corniculata it gave, the same photo. So probably their database is not that well trained, but Google has been having its own image search, and their search was much better, right? So when I used to Google it, find out that Sida cordifolia, which is closely related to this plant, Sida cordifolia. At least this gave a very wrong identification as Oxalis corniculata. So this is a very powerful tool. Now you can, this is a very clear example, even with a very poor photo, you can easily identify a plant here. And that is very useful tool for species identification. Nowadays, you don't need books. In those days, we used to use the Flora of the Presidency of Madras, the huge book, right? So nowadays we use this method, but this is a shortcut. I would not advise any of your students or anybody to use this method. It's better to use, huh?
They were not to be interested much for difference.
Yes. Because it's very easy, right? If you have this tool, you will lose that skill. That's the danger of AI. We are like how the students do the assignment also. They do it with the AI tools only, right? Like that. So they completely lose that skill there. And there are other databases also, like you have this leaf snap, where they digitize all the herbarium leaf shapes there. And then if you can, because identifying plants, the main challenge is it's very difficult, because unlike animals, plants are different at different seasons. Reproductive phase, they are different. Sapling stage, they are different. And mostly when you go, they are only in the leaf form. Or sometimes there's no leaf also, only branch will be there. So it's very difficult. At least 75 or 80, 90% of the times you will find them in leaf. But with leaf, we can't identify, because all the textbooks are helping us identify only with flowers. So these kind of apps help a lot. Leaf snap. But again, they also have limitations. This is only for the field guide, only for this US region, US, Maryland, and all Smithsonian. They have a leaf snap app for UK. We don't have this kind of app for India. We have to develop that. You know, the Pondicherry University has released a book on Western Ghats trees, how to identify based on leaves, just based on the leaves, because trees are very difficult. If you're walking, you will not find the tree. Even if you find a flower, it will be like 30-40 meters above. You can't go and catch the flower also. But fallen flowers would have been withered. Many of the parts have been broken. It's not easy to identify. So leaves are a better bet. Even leaves are above, but at least for fallen leaves, with the shape of the leaves, you can identify them. So you have to make a database based on the leaves. So this is a good app specifically for leaves. And coming to the next most important resource for plant identification is the plantnet.org. So here it is covering the entire world. It's also a community science program, or you can say community science venture here, where you can, when it comes to Indian subcontinent, it has about 7,472 native species, mainly for plants. So that's a very good number. By the way, how many plants are in India? How many species are in India? Does anyone know? How many species of plants are there in the world? 3.3 million, 3 lakh species so far, 3 lakh, around 3 lakh. In India, I think maybe it will be around little bit more than that. I'm not sure, probably maybe 50,000 may be too much. So that we need to find. In the world, we know. So there are 7,400 native species. There's a database for that. So here you type the name of the plant, it will come. But that search, what I did with Sida cardifolia, right? It took the photos from PlantNet only. Google searched the plant photos in PlantNet only, and then it came with a match. So that means all the plant photos are well documented here. So this is the PlantNet consortium. This is the paper talking about it. So this has been there for a very long time. It is a effort by the French people, many French. See all the French universities, French charities, they have come to this one. And this is a very good app, PlantNet. You can download it. I think many of the students also have this app, PlantNet. They can just click a photo and then it will identify it, like how the eBird and all were there. And then iNaturalist, again, is the most well-known, widespread citizen science app here. So it has, I think, these many observations, includes these many species, and it includes all animals, insects, everything, includes whatever biodiversity is there. So anybody can use it. When we also take photo and we become a part of it, we enrich the database. So I think this is a very powerful database nowadays. So even if all the specimens are there in the herbarium, we cannot go and access them. If it's in iNaturalist, we can access them very clearly. And you have the subject experts in each area, they come and validate it. And then they make the database better and better. And then, have you heard about this one? GBIF, Global Biodiversity Information Facility. So this is also a kind of large scale, large data project where the goal is to identify the occurrences of the species, where it's natively occurring. So if you want to study about a species, if you don't know the native range, what we have to refer the textbooks, right? That was the old time. But now you just type the species name. It will clearly show what all records it has from multiple databases. It also takes data from iNaturalist, but it also, the important thing is it takes data from the herbaria also. Herbarium sheets, musea, everywhere it takes the location where the plant or animal was initially collected. So this is very, very powerful resource. Everybody can use it for biodiversity analysis. And then you have this one, Gene Bank. Coming to Gene Bank, it has a lot of databases. We generally search for the nucleotides, any species, whether what genes are sequenced there. This is mainly used for molecular phylogenetics or molecular data. Now, I don't think how many accessions are there. I think it will be around a billion of accessions would be there. That many DNA sequences will be there. And you have all other sub databases also, including the genome also. Nowadays, many species genomes are also present there. And there's also information on NCBI taxonomy. They also give taxonomic information according to the current classification system. They will give, if you want to study one species, you want to know which genus it belongs, it can give you the classification system also. What are the other, based on the classification system, you can collect other genera which are present there. What genetic information, whether protein sequence is there, DNA sequence is there, anything you can download there. And when it comes to genome, so this is a very important resource nowadays for conservation biologists. My student, another PhD student, is working on conservation of a rare endangered species called Diospyros cruminata, a tree species. There are only 300 individuals in the forest itself. They are found only in Kerala, Karnataka. Maybe 300 to 500 individuals are only there. They are found